Check.
Wala. Ayun, umalis siya eh.
Meron pa yan, yung filters pa yan eh.
Sabi mo pa na, pabalik na po sa mga
Kasi yung display.
Ano pa ngayon?
Anubi, hindi nakasama sa cartoon picture. Si Anubi.
Wala ka?
Buti si... Hindi naman po siya
Kabila yung bago yung ginadad.
Hindi ko pa siya nakita na.
Hello! Pwede po ba bumalik na tayo sa halls for the competition? Hall A for professional, Hall B for non-prof.
Oy!
Hello! Kindly enter the halls again for the best paper competition. Let's settle down. Hall A for professional and Hall B for non-professional. Categories.
Hindi pala tayo pwede mag-chismis. Sige. Tapos after that, we can discuss it. Yeah. Kasi minsan mangyayari by yourself.
Wala nakikinig sa akin.
Wala.
Ayaw nila, sabi ko, pumasok na sila.
Sabihin mo, wala kayong snacks mamayang hapon.
Sisingkuhin ko kayo.
Ay,
Ano sabihin ko?
wala kayong snacks.
Sige, game. Sabi ko nga pumasok na sila. Para makakastart. Ano naman sila eh, nag-post house.
Correct. Kaya, yan yung aking suggestion.
Next time.
Next time, hindi pagsabayin.
Dapat naman.
Correct.
They have to ask about it.
Ay, 2:30. Sa taas.
Ako po na.
Bob?
Hello, once again. Kindly enter the halls for the best paper competition, which will start in 5 minutes. Thank you. Hall A for prof, Hall B for non-prof.
O yan ha. Nakailan ako. Ayoko na. Ikaw kaya ulit.
Hindi natin ano, no. Okay, yun sa akin. It's up at us. Parang relative ito eh. Kaya sabi ko nga, hindi pwedeng ibibigay natin kaagad.
Good afternoon, everyone, and Good afternoon, everyone, and welcome to the parallel session for Best Paper Competition Professional category. The Best Paper Competition is a scientific session that aims to recognize excellence in microbiological research. The research papers that qualify for the competition based on the preliminary and secondary screenings are then scheduled for final oral presentations. Today, we will listen to the final presentation of the 5 finalists from the professional category. The presentation of each finalist will then be followed by a 5-minute question and answer portion. Priority questions will be coming from our esteemed judges. For the audience on site, you may write your questions on paper that will be provided. For the audience online, please type your questions on the comment box, and we will try our best to accommodate all of them when time permits. To introduce the judges and to orient us on the criteria for judging, may we call on Dr. Pierre Angeli Vital, Division Representative for Aquatic Microbiology. Dr. Vital.
Wala.
Nakabukas.
Thank you, Sir Caleb. I am Pierangeli Vital, spelled without an E at the end of my name. Hi, everyone. Good day, and again, welcome to the Best Paper Competition Professional category. Before we begin the most awaited oral presentations, allow me to acknowledge the committee members, the Best Paper Committee Chair by Prof. Hannah Jane Dean, with members Dr. Lopez, Dr. Moron Espiritu, Professor Carlos, Prof. Pagoso, Prof. Cruz, and Prof. San Pedro, and myself. Allow me also to introduce our panel of judges who will evaluate the performances of our aspiring and youthful researchers. We are honored to have with us today the first member of the panel, Dr. Lynn Esther Ralios. Dr. Lynn Esther E. Ralios is a professor of microbiology at the Mindanao State University General Santos. She handles both undergraduate and graduate courses in microbiology and mentors undergraduate and graduate students in research. She currently leads research projects on antimicrobial resistance in animal-based foods and fungicide resistance of the Sigatoka pathogen in banana. And she also serves as a technical expert and reviewer for various external technical working groups, editorial boards, and consortia. Let us all welcome Dr. Ralios. The second member of the panel is Dr. Doralyn Dalisay. Dr. Dalisay, a pharmacist, microbiologist, and natural products chemist, studies microbial, marine invertebrate, and plant natural products for drug discovery. She earned a PhD in microbiology from the University of New South Wales and obtained a postdoctoral fellowship to study marine natural products chemistry at UC San Diego. Dr. Dalisay conducted drug discovery research at University of British Columbia, Vancouver, Canada, and Washington State University, Pullman, Washington, USA. She is the current and founding director of the University of San Agustin, also USA, for chemical biology and biotechnology. She is also a Philippine Academy of Microbiology diplomate. Let us all welcome Dr. Dalisay. And of course, last and definitely not the least, is the chair of the Board of Judges, Dr. Esperanza C. Cabrera. Dr. Cabrera is a full professorial lecturer 10 and university fellow of De La Salle University, Manila. She is also a fellow of the Philippine Academy of Microbiology and a fellow in public service of Metro Bank Foundation. She earned her BS Medical Technology degree from the University of Santo Tomas summa cum laude, MS PH in Medical Microbiology from the University of the Philippines College of Public Health, and PhD in Biological Sciences from the University of Santo Tomas. Her research interests are in medical microbiology, notably on the virulence and transferable multiple drug resistance of healthcare-acquired and community-acquired bacterial pathogens. Dr. Cabrera has been recognized for her teaching and research in microbiology with multiple awards. Let us all welcome Dr. Cabrera. Thank you to our esteemed judges for accepting our invitation to be the panel for this competition. Now back to the mechanics. Each presenter is given 15 minutes to present his or her research. The allotted time will be strictly implemented. Finalists, be mindful of the time placards. The placards will be shown there. Can you please show it po, yung placard, so that our contestants can see it? The placards will be 10 minutes, 5 minutes, 2 minutes, and time's up. Thank you, Ma'am Carlos. Extension beyond 15 minutes will merit a deduction from the total points. For example, 1 point per minute of extension. The presentation will then be followed by a 5-minute Q&A portion with our esteemed judges. Audience from on-site and online may still submit their questions, and if time will not permit, our finalists are requested to answer them via email. Criteria for judging are shown on the screen. Manuscript 60% and oral presentation 40%. The Best Paper Award will be given to the research that obtained the highest rating. However, a minimum score of 70% must be obtained to be considered to be a winner. In case of a tie, both papers will receive the award. The winner of the competition will receive a cash, a certificate, a plaque, and will permanently be a part of PSM history. A reminder that everyone is expected to practice proper decorum or etiquette before, during, and after the competition. The floor is now open for questions and clarifications before we begin. Are there any questions? Okay, if there are no questions, we can now proceed to the Best Paper Competition Professional category. The first presenter is Dr. Eloina S. Gampoy. Her paper is entitled SARS-CoV-2, Antibody Response and Serum Neutralizing Capacity of Early Unvaccinated COVID-19 Patients in the Philippines. Dr. Gampoy.
Hello. Hello. The clicker po not working. So, good afternoon, everyone. I'm Eloina Gampoy, and I will be presenting our study entitled SARS-CoV-2 Antibody Response and Serum Neutralizing Capacity of Early Unvaccinated COVID-19 Patients in the Philippines. So the coronavirus disease 2019 or COVID-19 was initially reported in December of 2019, and it has since become an ongoing pandemic worldwide. It is caused by the novel SARS-CoV-2, the severe acute respiratory syndrome coronavirus 2, which infects the host cells via attachment of the receptor binding domain of the spike protein to the human ACE2 receptors. Now, it's been shown that the spike and the nucleocapsid proteins are the main targets of the immunological response. So the main clinical features of COVID-19 patients are very heterogeneous and range from asymptomatic or mild presentation to severe outcomes. So the underlying factors for this heterogeneous presentation of disease severity have been an area of intense research, and our study is particularly interested in the humoral immune response of COVID-19 patients, which involves antibodies. Now, there are 5 different classes or isotypes of antibodies, each with their specific roles. And in our study, we are interested in IgG, IgM, and IgA because these are the ones mostly involved in the pathophysiological response of the body to the SARS-CoV-2, along with the different mediators of inflammation. So in the Philippines, although seroprevalence studies have been reported using commercial testing kits, there is still limited information on the reactivity of different antibody isotypes on SARS-CoV-2 spike and nucleocapsid antigens, as well as their serum neutralizing capacity, which is a better correlate of protection from viral infections. So briefly, neutralizing antibodies efficiently stop the infection by blocking the interaction between the SARS-CoV-2 virus and the host cell. And as it neutralizes the virus, it will promote viral clearance. So the information on the protective antibody response to SARS-CoV-2 infection has been studied thoroughly worldwide. However, antibodies can also exacerbate viral infection by different mechanisms. And additionally, many clinical manifestations of COVID-19 may be attributed more to an overactive inflammatory response. And theories on autoimmunity in COVID-19 have also been raised as the clinical and immunological manifestations of COVID-19 have similarities with the features seen in autoimmune diseases. Now, this link has prompted for the search for possible molecular targets of autoantibodies. And intuitively, one of the autoantigens widely studied is the ACE2, since it directly interacts with the SARS-CoV-2, and it is a major component of the renin-angiotensin system. Now, it is suggested that derangement in the renin-angiotensin system caused by the compromised activity of ACE2, influenced by the SARS-CoV-2 infection, promotes buildup of angiotensin II, and this arm promotes pro-inflammatory conditions that might contribute to the systemic inflammation that occurs in COVID-19. So taken all together, it is important to look into the role of antibodies, not just in protective immunity, but also to its contribution to disease severity. So almost 4 years into the pandemic, much research has been undertaken by the worldwide scientific community to understand COVID-19. Serological studies contribute in generating important epidemiological data for the control and prevention of infectious diseases, including COVID-19. And with the development of vaccines and emergence of different variants of the virus, the humoral immunity has considerably undergone dynamic changes. And our study intends to provide reference for comparisons with and interpretation of future epidemiologic studies assessing the antibody response in new coronavirus-related infection scenarios. Therefore, this study characterized the humoral immune response of unvaccinated COVID-19 patients in the Philippine General Hospital during the first wave of the pandemic, and this study was approved by the UP Manila Research Ethics Board. So briefly, biobank serum samples from COVID-19 patients collected from October 2020 to April 2021, prior to vaccine availability, were tested for the presence of binding antibodies against SARS-CoV-2 proteins and ACE2 receptor, as well as neutralizing antibodies. And then the levels of binding antibodies were then correlated with several clinical parameters. So first, we profiled the binding antibodies of patients with SARS-CoV-2 infection through the detection of anti-SARS-CoV-2 IgG, IgM, and IgA against spike-1, spike-2, receptor binding domain, and nucleocapsid antigens of the SARS-CoV-2 Wuhan strain, and detection of the anti-ACE2 IgG antibodies. So to perform the ELISA, antigens were first generated. So the nucleotide sequence of the SARS-CoV-2 antigens and the SARS-CoV-2 receptor were previously cloned into an in-house expression vector and transfected into HEC293 T cells. So the cell culture supernatant from the generated stable cell lines were collected and the respective proteins were purified. Lastly, positivity cutoff for the detection of each antigen-specific and isotype-specific antibodies were determined using true COVID-negative samples collected prior to December of 2019. So this figure showed the results of the anti-S1 IgG, IgM, and IgA among COVID-19 patients classified according to disease severity. So most patients, even those with mild symptoms, tested positive for anti-SARS-CoV-2 antibodies. However, the samples reacted differently depending on disease severity, and the mild group generally has a lower response compared to the rest of the patient groups. Also, most serum samples have higher IgM response than their IgG and IgA counterparts. And these trends are actually consistent with the other antigens. So for antibodies against S2, the IgG the IgM, and IgA responses in the mild group were significantly lower compared to that of the moderate, severe, and critical groups. For antibodies against RBD, the IgG response was significantly lower in the mild group compared to the moderate, severe, and critical groups. On the other hand, the IgM and IgA response were significantly lower compared to the moderate and severe groups only. Lastly, moderate, severe, and critical patients generally have higher anti-N IgG and IgM compared to the mild group. So in summary, most patients, even those with mild symptoms, tested positive for anti-SARS-CoV-2 antibodies. And this is necessary for developing protective immunity at the population level, as mildly symptomatic individuals account for the majority of infections in humans. However, the samples reacted differently depending on disease severity, and mildly symptomatic patients mostly have lower response than those with the severe form of COVID-19, which is hypothesized to be due to the differences in antigen exposure, as higher viral loads and prolonged viral shedding were observed more in severely infected patients. Also, most serum samples have higher IgM response than their IgG and IgA counterparts, as these samples were collected during hospitalization and are most likely in their acute phase of disease presentation, which is usually between 1 to 14 days post-symptom onset. On the other hand, IgG antibodies against ACE2 were detected in COVID-19 patients included in the study. However, no association was observed between anti-ACE2 IgG levels and disease severity groups. Next, we determined the neutralizing capacity of patient sera through a surrogate neutralization test, the RBD ACE2 binding inhibition, which is basically a competition ELISA setup, and a pseudovirus neutralization test. So first, a surrogate neutralization test was designed to incorporate a competition condition wherein the patient antibodies compete with the binding of ACE2 and RBD, and this provides a measurement of the potential of the patient's serum to inhibit RBD and ACE2 interaction. So a decrease in The ACE2 and RBD binding was observed when the competition assay was supplemented with the serum. And although the mean ACE2 RBD binding inhibition in each patient group was lower than 50%, the results still suggest that COVID-19 patients have antibodies that can reduce the interaction of host receptor ACE2 and SARS-CoV-2 RBD. Second, a pseudovirus neutralization test was designed to determine if patient antibodies can block the interaction of SARS-CoV-2 spike protein and membrane-bound ACE2 receptor, and then prevent the subsequent infection of the virus into the cells. So only 27 out of the 89 serum samples tested for pseudovirus neutralization test showed at least a 50% reduction against the spike pseudotype virus. So taken all together, the results of this study using 2 neutralizing measurements showed that a significant fraction of those initially infected with SARS-CoV-2 did not produce efficiently neutralizing antibodies, as was observed in previously published studies that reported variable neutralizing antibody titers in different patients in different countries during the early times of the pandemic. Lastly, we correlated the binding antibodies with inflammatory markers and clinical outcomes. So the serum markers of inflammation used in this study include the white blood cell count, D-dimer, C-reactive protein, and ferritin, which are routinely collected for COVID-19 patients in the hospital. And the patient outcomes include the hospital discharge or mortality. So this figure shows a summarized heat map of the Spearman correlation coefficient between baseline inflammatory markers and antibody responses to SARS-CoV-2 and anti-ACE2. And IgM was mostly correlated with inflammatory markers ferritin and CRP. And then most antigen-specific antibodies have a positive association with ferritin, CRP, and D-dimer. Notably, anti-ACE2 IgG antibodies that are hypothesized to be involved in a process that leads to inflammation was not positively correlated with any of the inflammatory markers tested. So therefore, antibodies against ACE2 detected during the time frame of this study do not appear to reflect an overactivation of the immunity function, but may have other purpose that merits further exploration. And only IgM antibodies against the nucleocapsid proteins was shown to be a predictor of in-hospital mortality both in the simple logistic regression and multiple logistic regression model. So in conclusion, these are the important findings that we were able to document in this study. SARS-CoV-2 specific antibodies were generally detected in hospitalized unvaccinated COVID-19 patients, and the antibody response profile varied in terms of antigens tested, antibody isotypes detected, magnitude, and disease severity. Patients here have low neutralizing capacity. The IgM response was associated with inflammatory markers, and only IgM against the nucleocapsid protein was shown to predict in-hospital mortality. And IgG antibodies against ACE2 were detected in the cohort. However, association was not observed with disease severity groups and inflammatory markers. So ultimately, the findings of this study provide a valuable baseline reference of SARS-CoV-2 antigen-specific and antibody isotype-specific immune response for possible emergence of related viruses in the Philippines. And in the event of such scenarios, the development of treatment strategies or drug discovery that can augment virus neutralization may be the focus, and those involving passive antibody therapies could be recommended and deployed with caution. So it is recommended that comparison to a more recent data in large-scale longitudinal cohorts be done to determine the evolution of changes in the antibody response of the population throughout the duration of the pandemic. And this will also enable to account the influence of the vaccination status and the circulating SARS-CoV-2 variants to the humoral immunity of the Philippine population. So I'd like to thank the following for making this study possible. So thank you all for listening.
Gampoy, for your presentation. We now open the floor for questions for Dr. Gampoy. We start first with our panel of judges. May I call on the chair of the panel of judges, Dr. Cabrera?
Thank you very much for the very clear presentation. How do I call you, Eloisa?
Eloy na, ma'am.
Eloy.
Yes po.
We are familiar with the auto antibodies IgG against the ACE2 receptor, which had been correlated with inflammation. It's said to be pro-inflammatory. However, as you have pointed out earlier, your results didn't support this. Like the level of your IgG against ACE2 did not correlate with the pro-inflammatory markers, ferritin and CRP, and the others as well. How may you explain this?
So the samples we included in the study are from hospitalization. So possibly the IgG levels are still not high. That's why IgG, anti-ACE2 IgG levels did not correlate with inflammatory markers, at least for acute presentation of COVID-19. However, anti-ACE2 IgG antibodies are also being suggested to contribute to the pathophysiology or the development of long COVID. So maybe if we extend the sample collection in chronic COVID-19 patients, maybe we can see a relationship between anti-ACE2 IgG antibodies and inflammatory markers po.
Just
Go
a
ahead, ma'am.
clarification, please.
Sure po.
That means that your results showed lower levels of IgG against ACE2 compared to the inflam markers?
There is no correlation between anti-ACE2 IgG levels and the inflammatory markers detected during admission of the patients.
All right, we move on po to Dr. Delisay.
Hello, Dr. Eloisa. So my question is, since we were able to your observation is that your IgM has increased on this sampling data that you have. My question now goes back to the clinical condition of the patient. Do you think that comorbidities influence the production of the antibody during your sampling? And that's the reason why those biomarkers of antibody that we expect to show were not shown.
Okay. So definitely the presence of comorbidities affect the immune response of an individual, and there is inter-individual variation for that context. However, unfortunately for this study, we did not account for the effect of the comorbidities or medications received, time to recovery, to the antibody response elicited in the patients.
Okay, thank you. Can I ask last
Sure
question?
po.
Clarification. You sampled 63 patients.
67 patients.
67.
Yes.
And yet you have a serum sample of 177.
77, yes.
Can you explain that?
Yeah, so for each patient, the serum collection or the blood collection coincided with the routine blood extraction for their clinical management. So these occur at days 1, 4, 5, and 7, 14 days of admission. So there are several patients who have completed the 5-point interval, and then there are some who did not complete the 5-point interval blood collection due to discharge or mortality outcomes. So for each patient, these serum samples are collected serially. That's why we have a total of 177 serum samples. But our sample size is only 67 patients po.
All right, let's have one final question from Dr. Ralios.
One final.
Go ahead.
Okay, I'll simplify it then, because actually my co-judges asked my major questions. I guess, alluding to what they have asked, no, do you find your results surprising or not surprising at all? And how would you, particularly your conclusion number 3, and as well as, the totality of your findings and conclusions, can you find, at this point in time, can you already find some practical application of the findings to the hospital setting?
All right, thank you. First, regarding the expectation on the results, so the trend that we observed, given that mild patients elicited lower antibody response compared to the patient groups with the severe form of COVID-19 are actually expected and are similar with previously published studies in other countries. And then for the second question, in terms of biomarkers, for the utility of this finding, at least for IgM, this is not routinely done in the clinical setting, or even IgG and IgA. Mostly these antibodies are used in the research setting for epidemiological studies. However, for the clinical utility of our findings, we can support that the use of convalescent plasma therapy should be recommended and deployed with caution, given that although recovered COVID-19 patients have high binding antibodies against the virus, these binding antibodies do not necessarily have the neutralizing capacity to the virus, and which is suggested to play a mechanism of action for the convalescent plasma therapy. So, there. Thank you.
All right. Thank you, Dr. Gampay, and congratulations. Another round of applause, please, for our first presenter. Insights on aeromodant phage diversity through physical, chemical, and molecular analyses. Ms. Gutierrez, please.
Okay, to start off, the abundance and diversity of bacteriophages is now well known. Phages is also known to have an influence in bacterial communities in various environmental niches. So with this, it is important to study the functional ecology of bacteriophages, most especially that they influence population control as well as evolution of bacterial immunity, metabolism, and lateral gene transfer. So these activities or natural activities of bacteriophages in the environment have presented a number of applications of phages as biocontrol agents and in phage therapy, as well as their role in biogeochemical cycles, bacterial evolution, and bacterial pathogenicity, which have been evident in the increase of studies and applications in the past decade. To support the recent biotechnological developments of phages, an impressive list of novel bacterial viruses have been isolated and characterized in other countries as well as in the Philippines from various sources, specifically through isolation of bacteriophages and testing these viruses in various settings from natural to artificial and industrial applications. So with this, I would like to proudly present to you our study in relation to phage ecology and diversity entitled On Diversity and Genetic Richness, Insights on Aeromonad Phage Diversity Through Physicochemical and Molecular Analysis. Again, I am Tracy Gutierrez, a member of the Bacteriophage, Ecology, Aquaculture, Therapy, and Systematics Research Group from the University of Santo Tomas. Okay, to start off, I would like to present rather some of the studies that have been related to phage applications in this country. So in relation to the therapeutic applications of phages, most of the research in the country are limited only to exploring the therapeutic applications of phages in medically important pathogens such as Pseudomonas, E. coli, and Salmonella. In relation to the target host of this paper, we have focused on the phages of targeting Aeromonas hydrophila. So previous published studies also show B614 and UP87 targeting Aeromonas hydrophila in induced infections of Aeromonas hydrophila, septicemia, and Oreochromis niloticus or Nile tilapia. So these papers present a promising result as they were the activity of the phages were comparable to that of a tested antibiotic, oxytetracycline. So the results show a decrease in the bacterial load in the blood specimens that was collected from the infected fishes, as well as a decrease in the mortality rate. So with the lack of study or limitations in the study in the country relating to phage characterization and identification, our study focused on sampling collection of and isolation of bacteriophages in sewage samples collected from Montreal, Canada, as well as in Manila and Batangas, Philippines. So after isolation, we focused on the physiological characterization of the phages, including pH stability, temperature stability, and morphological analysis. And lastly, we performed sequencing and phylogenetic analysis. The in-depth analysis of the genome sequences, however, is not included yet in this study. However, we will present to you our novel findings in the phylogenetic analysis in relation to the Aeromonas hydrophila phages. Okay, so in this paper, we did focus on the polyphasic characterization of these phages, focusing on the physiochemical characteristics as shown in this slide, represented by the morphological analysis, as well as the phylogenetic analysis using the protein-coded sequences from the phages that we have isolated. In brief, I would like to introduce to you the phages that we have isolated, which are 6 that were isolated from the Montreal samples or Montreal sewage samples, and 6 that were isolated from Lake Taal, Batangas, Philippines. In addition, we include in this study phages B614 and UP87. To start off with the physiochemical characterization of the phages, we proceeded with the pH stability, wherein the phage titers, or most of the lysates, were maintained after exposures at different pH gradients. However, the optimal infection of the phages was observed at pH 7. Additionally, most of the phages, or the most stable phages rather, that was isolated or studied in this paper was UP87 and B614. So the activities of these phages will be later discussed together with the morphological analysis and the temperature stability. Next, all of the phages exhibited lytic ability against the temperature gradient tested, with optimal activity or infection at 25 to 30 degrees Celsius. So again, the most heat-stable phages observed was phages UP87 and B614. So it is notable that compared to the other Philippine phages, as well as those that were isolated from Montreal, Canada, these 2 phages that were isolated from sewage samples in Manila, Philippines, exhibited the most stability compared to all those phages. So this can be also related to their original isolation source, which is the sewage isolated or obtained from a tropical country, compared to the sewage sample collected from a temperate country or Canada, or a pristine pristine water sample collected in Lake Taal, Batangas. The activity of the phages regarding its stability can also be related to its ability to aggregate. So in phage aggregation, the phages technically are basically trade off immediate infection for optimized production of the viral progenies. So in these cases, we should also keep in mind that the hosts can also be affected by the suboptimal conditions they are exposed to. So in this natural occurrences, it can be preferred that the phages do aggregate until such time that a suitable environment for both the host and phage infection is met. Last, for the physiological characterization of the phages, we proceed with the morphological analysis, wherein most of the phages that were characterized have a tailed morphology under the family or class Codoviricetes. Specifically, the phages or most of the phages that were isolated have a myophage morphology obtaining or containing a contractile tail, as shown in this figure. So this slide just shows the overview of the phages that we have isolated in the study, most of which featuring structures such as the capsid and the notable contractile tail, as well as the tail fibers, the collar, and the base plates. So these micrographs mimic or essentially correspond to the myophage morphology as shown in the diagram. So most of the phages in the study are myophages. However, they do exhibit, they do exhibit diversity in the sizes and the measurements in the structures as shown in this video. Most notably, phage DM12, phage DM12 exhibited the largest myophage morphology, resulting to a 260 nanometer measurement, while the smallest myophage isolated in this study was phage DM3. So based on this morphology alone, the different sizes in the diameters and the tail length show the diversity of the phages that can be isolated, although they are essentially clustered in the same morphological classification. So most of the phages that characterize have myophage morphology, and this reflects other studies that were done in Aeromonocytophila phages. So this includes the study by Bai and Rai et al., wherein novel phages were isolated containing the myophage morphology. Another morphology that was observed in this study was the podophage morphology, which is exemplified by the only isolate DM11. So this can also be reflected in the minority of the studies wherein podophages were also isolated for Aeromonocytophila. Okay, to proceed with the sequencing and phylogenetic analysis of the phages, so this phylogenetic analysis actually shows the reflection of the morphological classification of the phages based on their genomic sequences or genomic characteristics. So at this level, we can say that there are two major clusters that can be observed. The first bearing the podophage morphology under family Autography viridae, exemplified by DM11 or cluster 1. And the second group contains clusters 2 to 6, which is exemplified by the myophage morphology. So the discussion in the following slides is based on the species demarcation of 95% and genera demarcation of 70% for the phages as established by the ICTV guidelines. So for cluster 1, as I previously mentioned, this is found clustered under family Autography viridae, which is representative of the podophages. So with this, we obtain the similarity index of 89%, which corresponds to a new species, or suggests rather to a new species of phages under the septimovirus. Similar observations were found in cluster 2 and 3, wherein phage DM12 belongs to genus Syncasovirus with a similarity index of 88.4. So with this, based on the demarcation again, it can be suggested or assumed that DM12 is a new species under the said genera. For cluster 3, DM6 showed 84.2% of similarity index with the closest species under the genus Ishiguro-virus. So with this again, this suggests that the phage DM6 can be considered as a new species under the said genus. For the next three clusters, or the last three clusters rather, cluster 4 includes phages DM3 and DM4, which belongs to family Subreviridae and genus two-lane virus. So the species demarcation again that we have observed is 80% for both of the phages that were studied. So with this again, we can suggest that these two viruses, or phages rather, is a new species under the said genus. For cluster 5, which clusters phages DM1, DM2, and DM5, with a species or similarity index of 92%, it can be suggested that these two or these three viruses is also clustered as new species under genus Biquartaviruses. And then for the last cluster, which is have a similarity index of 62%, which based on the ICTV guidelines is below the genera demarcation for these viruses. So those that are clustered here, specifically DM7, DM14, B614, UP87, DM10, and DM9, can be considered a new phage species clustered under an undefined genus or new genus. So with this, the phylogenetic analysis and the morphological as well as the physiochemical analysis of the phages correspond to the varying degrees that a phage can be isolated and characterized, from preliminary identification through morphology to the classification of the molecular characteristics. So this degree of characterization and the polyphasic characterization of the phages may provide a level of understanding to the diversity and the nature of the bacteriophages. So this paper also serves as a first step toward phage ecology and diversity studies with the study of bacteriophages and the country still is in infancy. The data and the results collected in the study shows that there is a whole field of phage biology untapped in the Philippines. The authors would like to thank the following collaborators and institutions, and thank you very much.
Thank you, Ms. Gutierrez. We now again open the floor for questions. We start with Dr. Ralias.
Congratulations, Tracy.
Thank you.
Yes. So the study is pretty straightforward, but I'd like to be clarified on the heat map. Was it purely a qualitative type of assessment? And on your assay plates was the delineation of, because there are different scales, right? No lysis, just some lysis, so on and so forth, until you get to complete lysis. So is the demarcation there on your assay plates very clear for you to really rely on just qualitative assessment?
Yes, Dr. Reyes. Actually, the spot test that we performed generated this result. So from that spot test alone, we can already see the difference in the activities of the phages after exposures. We also did this test in triplicates so that the activity of the phages can be replicated and we can evaluate the results based on those replicates.
And the results across replications were consistent?
Yes, doctor.
Okay, thank you.
All right, Dr. Cabrera or Dr. Dalisay? Dr. Cabrera. Go ahead, ma'am.
This is not a question, but a comment. Wala pa ako sa question.
Okay po.
So can you please flash your heat map? Any of those, let's say temperature. For one thing, I really like to comment that this is a presentation, whether it's on the manuscript or the presentation.
Our judge is requesting to please project again the map.
Yes,
All right.
please. And
Thank you.
Can we stop the time while she's flashing she's looking for the table? Ang hirap ng 5 minutes eh. Okay. For one thing, I really would suggest, not suggest, but whatever table that you have, whether it's for presentation on a slide or in manuscript, you have to have a title so that can stand by itself. Another thing, if we were to look at whatever heat map that you have there, whether it's temperature or pH, the title of this is, let's say, temperature stability or pH stability of how many phages. But then if you were to look at this one, it is not so clearly represented. Like, for example, if you have that the darkest colored one, you have it lysis, complete lysis, etc. Right? But it doesn't make the reader understand it right away that you're referring to the lysis of the host cell, so that means that your virus is still active, your viruses are still active, and that means that they were able to withstand that particular factor. So probably it would help if, for example, lysis of the host cell, and that would now lead to the interpretation that your viruses were active. They were not inactivated at that part, something like that. Okay, my question is, if we were to look, sorry, wala yung sa 5 minutes, no?
Go ahead.
If we were to look at the proteomic tree, I will not, it was shown that the local phages were clustered together compared to those from Canada. And then it was explained that probably this would be due to a difference in the geography, like Philippines and Australia. But then if we were now to look at those from the Philippines, you have two different sources, lake and also sewage. I would like to know how they compare with each other, because their surroundings are, their environments are also so different from each other.
Thank you for the question and suggestion, Dr. Cabrera. Actually, we did analyze this further in an InPrep paper. But basically, we did analyze that there is a difference in their abilities or characteristics in the physiological level. However, during the molecular or genomic analysis, they do cluster together regardless of their isolation source as well as their activities. So this means that maybe the characteristics or the abilities, lytic abilities rather, of the phages is related to their nature. However, their in-depth analysis or essentially the molecular characteristics and the genome content of the phages, regardless of where they isolated, they can be clustered together and identified as such.
Dr. Delisay po.
Hello. Tracy.
Yes.
Tracy, hello, congratulations. Tracy, the title of your talk is on diversity and genetic richness. I was expecting a lot from it when I read your manuscript and the way you present. I was really hoping because the diversity and genetic richness that you presented are more qualitative. I was expecting more of a quantitative. And yung isa kong question, how come you weren't able to calculate the diversity index and the richness index of the phages that you were able to isolate? Because you already have the data, particularly in your molecular analysis. So I'm just wondering why such important index was not included in your paper.
So for this analysis, Dr. Delisa, I thank you for the question. For this analysis, Dr. Delisa, we only focused on the phylogenetic analysis of the phages as well as in its initial clustering. However, we did perform further analysis of the genome itself per phage, and we did compare it and perform hierarchical clustering. However, we did not already include it here in the paper since we did focus on the phylogenetic analysis and the clustering as well as the initial classification of the phages.
Okay. Yeah, the indices. So you have to calculate using the Shannon index and also for the richness index, the Margalef's index calculation based on the molecular data that you have. Next question for you. When it comes to ecological, ecology, microbial ecology, when we talk about diversity and richness, it should be very wide sampling scheme. And this is just for you to understand. You're very young as a student, or I don't know, master's, PhD student.
Done with masters.
Okay. So when we report diversity and richness, 2 sampling sites is not enough, or like 3 sampling sites in your case. So perhaps when you make this like a big paper and big program that you look into that, you have to go get different samples so that the diversity will be more comprehensive, and also the type of phages that you're going to get. So so good luck on more sampling and diversity of these phages. Thank you.
All right. Congratulations again, Ms. Gutierrez, and another round of applause for our second presenter. Let us now move on to our third presenter, Ms. Lordy Grace C. Itang, and her paper is titled Phenotypic Characterization and Complete Genome Analysis of a Novel Lytic Phage Infecting Pectobacterium Species. Ms. Itang, please.
Okay, good afternoon, everyone. It is with great pleasure that I will be presenting to you our study entitled Phenotypic Characterization and Complete Genome Analysis of a Novel Lytic Phage LET1 Infecting Pectobacterium sp. Bacterial phytopathogen causes significant loss of crop yield worldwide, posing threat to food supply. In particular, bacterial staph rat disease is widespread and is a destructive disease of vegetables and is considered as a perennial problem in the Philippines. The characteristics of the bacterial staph rat disease, it starts from the stem and also in the leaves and the underground part of the plant, hence causing these water-soaked translucent lesions. So it eventually proceeds to become softened tissue and becoming mushy or watery. So this disease causes a major post-harvest damage with annual losses of 15 to 50%. On the other hand, the genus Pectobacterium is part of the family Pectobacteriaceae, which is included in the important bacterial plant pathogens causing a wide array of plant disease. So these are gram-negative, facultative, and aerobe, and are non-spore forming. They produce a wide array of plant cell wall-degrading enzymes, pectinases, cellulases, and proteases, which are part of their pathogenicity. So, as to date, the complete eradication of soft rot disease is still not yet found. However, occurrence of the disease can be controlled or suppressed by the following methods, which includes the use of pathogen-free propagative materials, field inspection and eradication of diseased plants, and disinfection of cultivation equipment. Meanwhile, bacteriophages, as we all know, are viruses that infect bacteria. So in particular, the phage comes from the Greek word meaning to devour. Hence, the phages are referred to as bacteria eaters. So with 10 raised to 31 phages in the biosphere, bacteriophages are considered as the most abundant entities on Earth. So the phages may undergo the following phage lifestyle. So it may undergo lytic life cycle, where the phage will invade the bacterial cell, disrupting the bacterial metabolism, causing its lysis and productive infection. On the other hand, temperate phages may undergo lysogenic life cycle, where its genetic material is integrated to the host genome, where it persists as a prophage. With this, the bacteriophages has now gained renewed attention as an alternative biocontrol for plant disease, particularly soft rot disease, with their numerous potential benefits, which includes rapid degradation without host, low-cost reproduction, non-toxic, and with their narrow host range. Now, in this study, I would like to present to you that the novel lytic phage LET1 isolated in this study has a potential application for biocontrol of soft rot disease caused by Pectobacterium sp. Now, the objectives of this study includes to characterize LET1, a lytic phage infecting Pectobacterium sp based on morphological, biological, and genomic properties, to determine the taxonomic assignment of phage LET1 based on whole genome comparison and phylogenetic analysis of selected conserved genes, to evaluate the biological control potential of phage LET1 against Pectobacterium sp using tissue assays. So phage LET1 was isolated from symptomatic vegetable tissue and then was pooled for phage in SM buffer, and the phage stock was confirmed for presence of phage using spot test assay. It was enriched, and to purify the phage LET1, or to purify the phage, it has undergone a series of double layer plaque assay method until uniform plaque morphology was observed. So to characterize the isolated phage, high titer lysate was prepared using the confluent lysate method. So it was characterized based on morphological properties, which includes plaque morphology, virion morphology, biological properties, which includes host range, lytic activity, and optimal MOI, physicochemical properties, which includes stability assay, which includes the pH, temperature, and UV exposure. And also, the same high titer lysate prep was used for molecular characterization. So this includes polyethylene glycol precipitation of the phage virion to concentrate the plaque, the phage rather, and the precipitated phage was extracted for its genomic DNA. This was then sequenced using Oxford Nanopore technology, and then afterwards, the generated data was used for phylogenomic and phylogenetic tree construction. Now, for the results, phage LET1 produced clear plaques with turbid outer halos with an average diameter of 8.33 millimeters. Based on transmission electron microscopy, the phage LET1 is of a podoviral morphotype with a head diameter of 68.61 nanometer. Also, optimal MOI of phage LET1 was found to be 1. So optimal MOI is the optimal ratio of the phage titer in comparison with the bacterial load. Host range analysis of phage LET1. So host range, by the way, is defined as the span of genera, species, and strains that the phage can lyse. So out of the 89 SRPs, only 2 were lysed by phage LET1. Specifically, it's the host of phage LET1 and another Pectobacterium sp isolate 15b. And none of the 11 reference strains listed there were lysed by LET1. And none of the 5 Salmonella species multi-drug resistance isolate were lysed by phage LET1. So these results showed that the phage LET1 is a narrow host range phage. Phage stability test showed that phage LET1 is stable at pH 3 to 9 and was completely inactivated at pH 12. The phage LET1 is stable at a wide temperature range, and however, phage LET1 was inactivated 5 minutes post UV exposure. So this is quite important for field application since the temperature in the field does not exceed beyond 50 degrees Celsius, and the pH level in the field is not optimal pH for plants, especially is around pH 5 to 6, and for the soils around pH 3 to 3.4 to 4.4. Now let's proceed to the complete genome analysis. Phage LET1 has a circular double-stranded DNA with a total genome size of 40,519 base pairs, a GC content of 52.3% with no putative tRNAs. It has 50 predicted coding sequences, 26 of which has putative functions. Also, it is interesting to note that there are genes for lytic activity observed in this genome of phage LET1, which includes the endolysine, arzylike lysis protein, and choline. And no genes for lysogeny were observed. Comparative genome analysis of phage LET1 showed that it has 89.36% similarity with 81% query cover to Enterobacter phage IME278, which is an unclassified virus, 85% similarity with 81% query cover to Salmonella phage PRF-SP2, which is also an unclassified virus, and 92.52% similarity with 79% query cover to Shigella phage SFP21, which is unclassified virus with sequence divergence of 50 base difference for every 100 bases. Now, based on the species demarcation by ICTV, species demarcation is set to 95%. Now, aside from comparative genome analysis, owing to the mosaicism of the phage and lack of universal markers, utilization of open reading frames for conserved gene phylogeny were used for this study. So this includes the major capsid protein and terminase large subunit. So for the conserved gene phylogeny, the major capsid protein forms the icosahedral head of the phage. So phylogenetic analysis showed that it has 97.13% similarity with 100% query cover to Escherichia phage IMM002, which is a kyphanovirus, and has sequence divergence of 2 base difference for every 100 bases. The other conserved gene is the terminase large subunit, which translocates the phage DNA for packaging to the prohead. So based on this result, terminase large subunit of LET1 when aligned with the other 50 sequences of terminase large subunits, it has 98.26% similarity with 100% query cover to Shigella phage SFPH2, which is a kyphanovirus, and it has only 45% similarity with the other phages shown there with no sequence divergence. Now, other genes for phylogenetic analysis, these are genes related to the lytic activity of phage LET1. So these include endolysine and RZ-like lysis protein. Choline was not included here. So for endolysine, it has 94.08 similarity with 100% query cover to Enterobacter phage EP1, which is an unclassified kyphanovirus with sequence divergence of 5 base difference for every 100 bases. And the RZ-like lysis protein, which is also genes for lytic activity, it has 94.63% similarity with 87% query cover to Shigella phage as of PH2, which is a kyphanovirus, clusters together with Enterobacter phage EP1 and classified kyphanovirus, and Escherichia phage RDN8.1 and classified kyphanovirus with sequence divergence of 2 base difference for every 100 bases. Now, with this result, we are proposing this classification for phage LET1. So it is under the realm Duplodnoviria, which includes all viruses with double-stranded DNA coding for HK97 fold major capsid protein, kingdom Hungangvirae, phylum Uroviricota, which includes all viruses coding for the terminase large subunit, the class Codoviruses, which includes all tailed bacteriophages, order Autographiviridae, which means that they are self-transcribing viruses, so they encode their own RNA polymerases. Basically, phage LET1 has its own RNA polymerases in the supplementary material. Family Osteodervirinae. Interestingly, this family also includes the lytic Pectobacterium phages from the study by Evziv et al. 2020. They are also a podoviral morphotype with icosahedral head of about 16 nanometers, genus Kyphanovirus, and species unclassified Kyphanovirus. Now, lastly, biocontrol of phage LET1 was also studied. So here, it is shown that at MOI 10, when tested in cabbage tissue slices, the phage LET1 caused reduction of about 56.32% only. However, at MOI 100, it causes a disease reduction of 88.51%, which means that at higher MOI and at higher titer, the phage is effective at controlling the soft-rot disease. So the highlight of this study are the following. So this study describes the isolation, characterization, and potential application of LET1, a lytic phage infecting Pectobacterium sp. LET1, which is a non-tailed phage belonging to genus Kyphanovirus, produced clear plaques with turbid outer halos and was found to be stable over a wide range of pH and temperature conditions. And phylogenomic and phylogenetic analysis strongly suggest that phage LET1 is a novel phage. Lastly, phage LAT1 exhibited strong lytic activity against Pectobacterium sp in semi-implanta assays, suggesting that this novel phage may be a potential candidate for phage-mediated biocontrol of soft rot disease. Now, I would like to thank the collaborators for this study and institution. Thank you.
Thank you very much, Ms. Itang. Now the floor is open for the questions, starting with Dr. Delisay.
Hi, good afternoon, Lordy.
Good afternoon,
Another
Doctor.
phage story that you presented to us. So my question is on this aspect. The reason why you have the phage is because of the plant, because of the bacterium infecting a plant or a vegetable or a produce. And I was really wondering, why did you check other properties for its lysis to those organisms that does not infect plants? And those organisms are human pathogens. And what's the logic there? Are you just trying to look into its lytic potential, that it can be a broad spectrum type of virus?
Thank you, Dr. Delisa, for that question. So one is we tested other bacteria aside from those SRPs because we want to establish that the phage is a narrow host range phage. And also, basically, we want to just establish that it is a narrow host range phage and it does not lyse other bacteria of different genera.
So is that of advantage to crop protection later on? Is a narrow range virus is an advantage for crop protection later on?
I believe, Dr. Delisa, yes, it is an advantage because it does not affect the natural microbiota of the plant, except for the bacteria that is causing the disease on the plant.
Okay. Can I ask a last question?
Sure, go ahead.
So the novelty, you said you have this phage that is novel. When we talk about novelty, sometimes like people say it's a novel, but actually it's not kind of a novel. So we have to be very careful in using novelty in the data that we have. And so what criteria did you base the novelty classification of your work?
We base it on the species demarcation according to ICTV, which is 95%, and also based on the complete genome comparison, phylogenetic analysis, phylogenomic analysis. It only has around 92 point something percent in comparison to its closest phage, which entails that possibly this phage is novel or is new under the virus.
Okay, I suggest do more of the genome mining, particularly on the lysis gene. And then compare it to those that are broad spectrum and then see if you can see more novelty there rather than looking into just like the whole genome and the demarcation that is 90% to its next similarity in the phylogenetic tree. Thank you.
Thank you for that suggestion, Dr. Delisa.
If I may, I have two
Dr.
questions.
Cabrera,
the
go
first
ahead po.
one is related. Because like the title says, it's a novel phage. So you had your percent similarity and also, but these had low query covers. So when compared to Enterobacter phage and also your Shigella phage, aside from that, you also had your phylogenomic tree. So all of these were based on the results of your nucleotide sequencing that made you say, oh, these are quite low, so this is novel. My question is, how robust are your BLAST results? You subjected it to BLAST, and they come up with so many parameters. Among those different parameters, how robust are your results to say that this is not due to some technical errors or I need more sequencing? Because how many times was this also sequenced? You know that when something is claimed to be novel, it takes more than just one. I know you came up with a consensus sequence, right? But then how many times was this sequence, and how robust are your BLAST results?
Actually, thank you for that question, Dr. Cabrera. Actually, it was there in the table for the, although I did not mention, but the coverage for the when it was sequenced using the ONT is quite high. I cannot actually remember the actual value, but it was there in the table.
Are you referring to the genomic sequences?
Yeah, yes, doctor. It
You had, I don't have the figures here, but
it
it's
was
like
there,
you,
but
you
I
only
cannot
have
actually
89.36.
Yes.
89.36 similarity to enterobacteriophage, and
The-
then, but the query cover was 81%, etc.
Yes, but the when it was tested, the mean coverage was, the actual value is, I cannot specifically recall the actual value, but the mean coverage was mentioned or was
Maybe
flashed.
you have it in your manuscript or in your slide. You want the slide to be shown?
Yes,
The
please.
technical team, please help put up the slide again for Ms. Itang.
While it is being
Hello.
set up, may I?
Sure, ma'am, please.
So you tested the host range, and then your LET1 phage was able to lyse only 2 out of 89 of your, what's this
SRP.
genus name? Pectobacterium. So will
Yes.
that be nice to use? Will it be an effective agent against Pectobacterium? When it was able to lyse, it showed activities only on 2 of the 89 isolates tested.
I think it would be beneficial. However, if there are more studies on those other Pectobacterium as species in our collection, it could be used in tandem together with the phage that I have as a potential biocontrol agent for the Pectobacterium species that we have in our collection that causes concerted infection on the plant, because the disease is not just caused mainly by either one or two. It could be made up of several pectobacterium. So if there are more isolated phages that could also lyse those Pectobacterium species that we have in our collection, it could be used together in tandem with the phage that I have isolated.
But my question is based only on the results of your study, and you claim that this could be a good one, good agent for against Pectobacterium. But then the results are not so encouraging as far as its host range is concerned.
Yeah.
So, thank you. I'm referring to the genomic results, the genome sequence blast, not particular genes.
Yes, the
Yeah, the question is, what made you conclude that you have a novel phage isolate?
actually only the top 50 phages among the results were shown here, and so it shows that it has only this similarity. I failed to mention earlier that the And the mean coverage of the O1 sequencing was 12,885 times. And where was this one? So this was tested against the top 50 among all other phages in the database. So only the top 50 were shown here. And that's the result, 89.36% similarity, 85.
Okay. Thank you, Ms. Itang. I think we have to move on. Okay, one final po, ma'am, please.
One final. Oh, I have many questions for Ms. Lordy, but congratulations, Lordy. Yes, one, just quick, please allow me to. Very quick.
Okay.
And please
Okay.
answer quickly. The first is related to the question on the, the rationalization behind the choice of isolates to include. But my question, I think you answered already on the part of the test isolates for the lysis assay. But my question now is, why did you select strain 2CAB3 2/C15? Is it the most virulent in your, in terms of rot in your collection, or what was the reason behind? Just answer quickly so I can ask a second question.
Thank you,
Maybe one concise,
Thank
full
you, Dr.
sentence.
Reyes. Actually, 2CAB was not the only soft rot that we soft rot disease-causing bacteria that we have in our collection. However, that was the one that was passed on to me. We have a separate group studying on the bacteria alone. So I am on the phage part. So that's the isolate that was passed to me. So I was focusing on that.
Okay, but anyway, just a comment. I'm sorry, I can't help but comment. Because you're right, usually for plant diseases, no, common yung complex. Now, diseases are usually caused by complexes. And so, yes, you might want to consider looking at different, not just one phage strain, yes, but keep looking to come up with some kind of a consortium to hit. Okay, so but anyway, my second question, if you will allow me,
Go,
Dr.
ma'am, go na
Pierre,
po.
is the intention of this research is for biocontrol, right? So my mind was already running to the field when you were talking about this. So my question for you is, what would be your major considerations in testing phage LET1 or maybe future phages that you will discover under field conditions to bring this study closer to commercialization?
Thank you, Dr. Alias, for that question. Now, if we put our mind to the commercialization of these phages, the first thing that I would like to consider is the stability of the phages, because not all phages will have the same stability in terms of several parameters like the pH, the temperature, even its UV exposure. Now, for this phage, since it was inactivated 5 minutes post UV exposure. So although this was tested against UVC radiation, so I would like to recommend further to test these phages for its stability on UVA and UVB, since those type of UV are more commonly found in the field, or the plants are most commonly exposed to this kind of radiation. Also, since the phage is quite stable at a wide range of temperatures, so this is not a problem at all for this phage. And for its pH, for example, its pH stability, since it was inactivated at pH 12, so we should consider that it's this solution where it will be suspended should be within the pH range where it would most likely be viable, and the titer will not be reduced before it will reach field application.
All right, congratulations, Ms. Itang. Another round of applause, please, for our presenter. I am very sure that you still have other questions, so please keep them coming. Just send them in, and our presenters will answer them through email. Let us now move on to our fourth presenter, Mr. Angel Bert D. Cortez. His paper is entitled Indigenous Pectinolytic Bacteria as Potential Starter Cultures for Coffee Fermentation to Improve the Cup Quality Profile of Coffee. Mr. Cortez, please.
Okay. Good afternoon, everyone. I would like to present to you this project entitled Indigenous Pectinolytic Bacteria as Potential Starter Cultures for Coffee Fermentation to improve the cup quality profile of coffee. This project is actually part of the USDA program implemented by the ACDI VOCA through their PhilCapi project in the Philippines. And this is in collaboration with the Barista Coffee Academy of Asia and Equilibrium Intertrade, so more in focus on coffee industry. So again, my name is Angel Bert and I am from Cavite State University. So we know that coffee is a viable agricultural commodity that makes economic and social impact worldwide. Based on this map, you can see that many countries or coffee-producing countries in the heat, or let's say in the coffee belt, are gaining much interest in the coffee production because it actually produces an income for them. So nowadays, the coffee consumption has been increasing and it will be increasing in the next years. So in the Philippines, according to the Philippine Coffee Industry Roadmap, we are producing 4 types of coffee. And based on the production of green beans, Robusta gains the highest production, followed by Arabica, and then Excelsa and Liberica. So in the Philippines, the consumption of coffee has also been increasing. However, we don't have enough supply to suffice those consumption. That's why the Philippines is just 15% self-sufficient in coffee. So the problem here is not just about the production of cherries, but of course, the production of the high cup quality profile of coffee, particularly with Excelsa and Liberica, which are also considered as Kaping Barako, which has a very earthy taste as compared to Arabica and Robusta. So here in the Philippines, since the Liberica Excelsa are commonly cultivated in Southeast Asia, and in particular in the Philippines, we want to improve the quality of this coffee that will somehow compete with Arabica and Robusta soon. Okay. But before that, okay, so specialty coffee are gaining much attention to the coffee consumers because of the a lot of available specialty coffee in the market. So the cup quality profile of coffee is usually associated with the post-harvest management operations that determine the quality of the coffee, and this includes the fermentation method. So the fermentation plays an important role, or let's say particular in the wet processing method, because this one is commonly driven by the microorganisms, particularly in the degradation of the coffee mucilage, also inhibition of the mycotoxin-producing fungi, and of course, the production of the flavor-active compounds. So as I've mentioned, the fermentation is due to the interplay of the microorganisms at the same time, the bean metabolism. So the interplay of these microorganisms and the bean metabolism affect the cup quality profile of coffee. So many organisms can be isolated and studied during fermentation, and one of those are lactic acid bacteria, acetic acid bacteria, and even those Enterobacter, members of the Enterobacteriaceae. And those groups can be lowered down into pectinolytic bacteria. So these bacteria are capable of degrading pectin in the coffee mucilage. If you look at this coffee cherry anatomy, one of the layers of the coffee cherry is the pectin layer. So the pectin layer is really hard to degrade just using water, because it's made up of a very thick polysaccharide substance, and that's why microorganisms taking important role in the degradation by producing this pectinase enzyme. So the pectinase enzyme has the capability to degrade those pectinaceous substance, and once it happens, the bean or the green bean production will be accelerated. So the objective of the study is actually focused on the starter culture development. So we know that starter culture will accelerate the fermentation process, and it reduces the acidity and therefore increasing the production of the green beans. So the objective is, of course, to isolate pectinolytic bacteria, and these bacteria can be isolated from those enumerated total aerobic bacteria. And then after that, we will characterize them. Aside from the pectinase production, we also characterize them in terms of the production of the amylase and also the protease, and their tolerance also to the important fermentation abiotic stresses. And lastly, of course, we want to know their identity, so we subjected them to molecular identification. So specifically for our methodology, we collected the cherries in Itogon Benguet. So we collected the isolates or the cherries from Itogon Benguet, since Arabica will only grow in high elevated areas. And then we subjected them to fermentation process, so for 48 hours, and the samples were collected 12-hour interval. And then we enumerated the bacteria, particularly the total aerobic bacteria, and there we get colonies for screening of pectinase production. Then we subjected the isolates for screening of the pectinase production using the pectin as their substrate for their medium growth. And then lastly, second to the last is we also assay their enzyme production by allowing them to express this hydrolytic enzymes such as protease and amylase, and of course assay the tolerance to acidity, salinity, and alcohol concentrations. And lastly, we also subjected them for molecular identification by targeting the 16S RNA gene. Okay, for the results, these are the environmental factors that we have analyzed during the fermentation process: the pH, the temperature, and the total aerobic bacteria. So in terms of the pH decreases as the process progresses, and the lowest pH was observed at 24 hours, gaining the pH of 4.024. However, the temperature remains constant, of course, because it's a controlled environment. And in terms of the population of total aerobic bacteria, there is an abrupt increase in the first 12 hours of fermentation from 6.95 times 10 to the 5 CFU per mil to 3.21 times 10 to the 7. And the total aerobic bacteria at 24 hours is at highest maximal rate of 7.05 times 10 to the 7 CFU per mil. Okay, in terms of hydrolytic enzymes, these figures shows the assay for the pectinase, the amylase, and protease production of the isolates. Those figures in A, B, C are the controls, so those isolates don't have activity on the medium, but for the D, E, F are those with activity. So for letter D, that is an isolate having this clear halo zone around the colony, indicating a production of the pectinase enzyme. And at the same time, for letter E, it is production of amylase when we grow the colonies on the starch agar medium. And lastly is for the protease. So we use here UHT, non-fat milk. So there is a degradation of casein. So this is the table showing the solubilization index of different isolates when we subjected them for different assays. So for the pectinase, we extended it for 96 hours because, again, the pectin is really hard to degrade. And based on our observation, so we already got a result after 96 hours. So we suggest that the production of these enzymes, the hydrolytic enzymes, implies a promising potential as bacterial starters for coffee-related biotechnological industrial applications, since, of course, the degradation of the degradation of the mucilage in the coffee involves various enzymes, such as those pectinase, amylase, and of course, protease. Now, we also subject them for tolerance to important abiotic stresses during fermentation, such as acid, salt, and alcohol tolerance. So you can see that all of the isolates were able to tolerate the concentrations of alcohol up to 10%. And for the salt tolerance, isolates P5TA3 and P3TA1 has minimal growth in 3%, but majority has or have tolerated the salt concentrations. And then for acid tolerance, all of the isolates have tolerated the pH 4, but this isolate P5TB5 has exceeded and tolerated a 3.5 pH. So with this ability of the bacteria to develop tolerance to these multiple stresses during fermentation can be attributed to the ability of these isolates to become homeostasis or to maintain the pH homeostasis, and also the stability of the membrane, the fluidity, and metabolic regulation and macromolecular repair during the process. And lastly, in terms of the identity, so this shows that all these isolates are gram-negative and rod-shaped, and then most of them belong to the Enterobacteriaceae, which is given because based on literature is one of the groups of organisms that can tolerate during fermentation or propagate during fermentation are those members of the Enterobacteriaceae. So we have identity based on a 16S RNA gene, the Crassiobacterium, Entelagicum, members of the Enterobacter, Serratia, Klebsiella, Citrobacter, and Leclercia. So the fermentation affects the metabolic activities of these natural microbiota, which predominantly grow and uniquely impact the coffee quality. However, upon observation, some of these isolates are pathogenic, and like the Klebsiella vericola and Enterobacter cloacae, but based on my research, these organisms really exist in coffee fermentation, so we cannot really avoid them to being present there. So the study successfully screened pectinolytic bacteria with protease and amylase together with their tolerance to abiotic stresses. And these bacteria, based on the analysis, belong to the Citrobacter, Crassiobacterium, Enterobacter, Serratia, Klebsiella, and Leclercia genera. Now, the pectinolytic bacteria having this potential to be used as certicultures during coffee fermentation. So the coffee fermentation can be optimized using this bacteria to speed up the processing time, improve the cup quality, consistency, and reduce the post-harvest failures such as those caused by contamination and other damages. So since the coffee is seasonal, we are still waiting for the coffee cherries to be available so that we can test these bacteria for the effectivity to really increase or improve the cup quality profile. We also have isolates of lactic acid bacteria and acetic acid bacteria, and we are planning it to have a consortium during the application process. So with that, from Cavite State University po, tara magkape. Thank you.
Thank you, Mr. Cortez. Tara, kape na po tayo. Sige po. Let's have, again, questions from our Board of Judges, starting with Dr. Ralias.
Thank you for giving me the the microphone first. Congratulations.
Thank you, ma'am.
Your name is?
Angelbert po.
Engelbert. And I do love coffee. So as to your isolation, I think I did not see even in your paper, how many unique colony morphologies did you obtain on your PCA? And did you also consider, we all know there are many non-culturable but viable microorganisms, and these are probably giving more impact on the beads, on the fermentation process itself, than the minor. I say minor because usually, di ba sabi natin 1% lang ang kaya natin cultivate. So did you also consider the contributions of those non-culturable, at least in the back of your head? But anyway, how many unique colony morphologies? Was that very diverse? And why was salt tolerance included in the characterization of these isolates? Does salt tolerance have an impact on the fermentation process?
Okay, thank you very much, Dr. Ralios. The first question is how many isolates I picked. I picked 35 colonies based on their differences in colony color and other characteristics. So not much, ma'am. And that is based on differences. So although it also includes other, it came from other medium, but from that PCA, I only collected 35. And out of those 35, only 10 has positive results for pectinase production. And then for the next question about the salt tolerance, thank you for that. For the non-culturable, ma'am, we have a proposal on determining the profile of community during the fermentation, but I wasn't able to include that in my objective, ma'am, to know the effect of those non-culturable bacteria. But that's in the pipeline. Yes, ma'am. We have actually a proposal undergoing, And we want to know those microorganisms that might also play a role during the fermentation process, particularly if it's already applied or the starter cultures have been applied. So we want to know the correlation and association. Hopefully the funding agency would approve that. And then for the salt tolerance, I am a certified capper po, and we cup coffee based on the aroma, fragrance, and one of the characteristics is determine the saltiness of the coffee. And the salt concentration is very important because once the organisms were able to tolerate certain concentrations of salt, then it has a contribution onto the effect, particularly during the cupping. Because our goal here is to make the the Liberica Excelsa to be specialty coffee. It means that it has a certain grade to become 80%, and once it's 80%, it's considered as specialty, which is commonly observed in Arabica and Robusta. And again, one of the categories there is to determine the saltiness of the coffee, and that's the reason why I included that in the process.
So just clear, so the saltiness actually has an effect on the
Quality,
flavor.
yes, yeah.
Okay.
The quality overall,
so that's a
ma'am.
good learning for us today.
The quality overall of the coffee, since we have the aroma, taste, fragrance, what else? The off flavor and so on, and then you can get the percentage of 80. So once you've reached that, it means that the coffee is specialty coffee.
All right.
Thank you po.
Maybe Dr. Dalisay would like to go next.
Hello, Angel Bird. I'm not a coffee drinker, but I like the aroma of that. So this is just one comment. I was kind of like misled by your title when I was reading your manuscript last night. I was expecting after the
Application.
isolation of bacteria, then you have improved it in flavor, aroma, color, taste, and everything, but it was not included in
Yes.
that. So bitin ako, right? So that's one. So my question, from the different varieties of coffee available in the Philippines, why did you choose Arabica as the source instead of Robusta or Liberica and Excelsa?
Thank you, ma'am, for that question. Our funding agency prioritizes Arabica, and we need to, what do you call this, fit the isolation from the Arabica itself because we wanted to apply it to Arabica. But of course, as a researcher, we need to be flexible enough to also consider other varieties. And after this, ma'am, we will be considering application of the isolates, not just in Arabica, but also in other types of coffee. At the same time, Arabica gains in terms of flavor, they are the ones that have this very aromatic, fruity, flowery scents. So maybe microorganisms in that particular beans or cherries have the contribution, and we wanted to isolate them. Maybe that if you can isolate them and apply them again, if the cup quality has been improved, then maybe they are the ones that contribute to that quality coffee of Arabica.
Okay, thank you. So another thing, I'm kind of like confused of what really is the goal of your work.
Yes,
So
ma'am.
you took a cherry bean and then isolated bacteria out of it.
Yes, ma'am.
So you have the bacteria from this coffee, and then my question is, if there will be no starter culture, can this cherry bean can undergo fermentation by just putting all the cherries together and let it ferment?
Yes, ma'am. It's basically it can ferment. So the
By
goal of a
itself.
starter culture, sorry.
Yeah, by itself.
Yeah, it
Yeah.
can ferment.
So I think you need to be very clear on your objective. That your objective is to create a cocktail of bacterial starter culture that can be to ferment a coffee, right? So on that context, are you looking into a cocktail of everything that you have isolated, or just one or two bacteria?
Okay, thank you, ma'am, for that. So based on the results, some of those are red flags like pathogens. So basically, we will not consider them. However, since we have a lot of isolates, we will be trying to, ideally, it should be applied one by one. But if you are, according to the coffee expert, the ones that they buy in the market is actually a consortium of bacteria. So maybe just to include them all, we can make a consortium and apply them as one in the coffee fermentation.
Yeah, because the microbiome of fermentation is so rich
Yes,
that it is a community of diverse organisms there. And taking and applying just one organism for fermentation, I do not think that it could be beneficial to create these different types of quality product that you can have. And just like chocolate, there are hundreds of inoculant, a microbiome present in the fermentation. But I like your project, and good luck on that.
Thank you so much.
All right po. One final question from Dr. Cabrera.
Yeah. Comment first, not a question. Comment first. I have the same comment because I was about regarding the title.
Yes,
I
ma'am.
was expecting more because
Yeah.
it sounded so romantic to improve the cup quality profile of coffee. So bitin nga, ano? So my suggestion is this is a scientific paper,
Yes.
not a literary work, so we can make the title more accurate, or
Yes,
not just more accurate, make it accurate.
Accurate.
My question is, you presented several solubilization indices. Are there standards for the interpretation of these? Because it was not just a matter of plus, minus. You had measurements, but there was no interpretation of what these mean. Are you supposed to do stat analysis to compare one with the another? I'm not familiar with that.
Thank you for that, ma'am. We did have a standard for solubilization. However, we have formula to get the index. Now, in terms of statistical analysis, we did not perform stat analysis, ma'am, because we just confirmed that they have activity. But we also included the index based on the value that we get on the plate.
Okay. No, this is another question, just a clarification on the answer. So, in this case, because you were saying that there are solubilization indices standards?
No, ma'am.
Wala. So, how do you treat these data?
We just use it like the activity is present or not present in terms of solubilization. But we've included the measurement po, just in case if you want to know the index.
Nothing that would say this is promising, so work on this or junk this.
Yes. We have another test to determine the quantity of the enzyme produced. We're going to use spectrophotometer. Actually, we have a data, but because of the deadline, we're able to include it in the paper. Yes, ma'am, we have a result on the pectinase production, using the spectrophotometer analysis.
Okay po. Okay na pa. Ayan. All right. Congratulations, Mr. Cortez, and another round of applause, please, for our presenter. We move on to our last, as we are pressed for time. The last presenter, and definitely not the least, is Mr. Bonnie B. Datul with his paper titled First Report on the Gut Microbiome of the Endemic Greater Musky Fruit Bat, Tenochirus jagori, Peters 1861, from the Cavinti Underground River and Caves Complex, Cavinti, Laguna, Philippines. Mr. Datul, please.
Thank you, Dr. Pai. Good afternoon, everyone. I am Bonnie Datul, again, of UPLB Museum of Natural History, and I'd like to present to you our paper entitled First Report on the Gut Microbiome Profile of the Endemic Greater Musky Fruit Bat from Cavinti Underground River and Caves Complex in Cavinti, Laguna, Philippines. So this paper is part of the Study 1, Comparative Microbiome Studies of Bats and Bat Guano present in caves in Calabarzon, which is one of the studies of Project 4 of the NICER program, Center for Caves Ecosystems Research in Calabarzon. So this will be the outline of my presentation. So recently, just this May 10, 2023, during the 42nd ASEAN Summit, the Association of Southeast Asian Nations established the One Health Initiative. This was in response to the devastating and multidimensional effects of the COVID-19 pandemic, as well as the emergence and re-emergence of infectious diseases such as zoonosis. Indeed, COVID-19 have focused the attention of the public to the interactions between humans and its environment, especially on the transmission of infectious agents from wild animals to humans. COVID-19 especially shifted the focus to bats. And so bats are the second largest order of mammals. They are almost located everywhere, and they are well recognized as natural reservoirs of vectors of microorganisms and viruses, some of which causes significant pathogenicity to humans. And bats have a number of ways to transmit pathogenic or infectious agents to humans. First of which is through contact with body fluids such as urine, feces, or saliva. Another transmission is by wild animals that has bat-borne viruses or bacterial pathogens, such as the Ebola virus. Natural predation by monkeys and domesticated cats can also be a mechanism for transmission of infectious agents from wildlife animals to humans. We actually observe a natural predation of a domesticated cat catching a flying bat while we were on a field work in Rodriguez, Rizal during one of our field works. Another mechanism of transmission is through bat ectoparasites. Bat ectoparasites such as ticks, mites may incidentally bite humans that are encroaching on cave ecosystems, and this may cause transmission of infectious agents from bats to humans. Finally, environmental transmission through contamination of habitats used by infected hosts has been recognized as a mechanism of transmission of infectious agents from wildlife animals to humans. So the Tenochirus jaegori, or the greater musky fruit bat, is an endemic fruit bat to the Philippines. It is a common and widespread species, and sometimes it encroaches into urban areas. It also tolerates degraded habitats, and one of the major steps, or one of the essential steps to the control of infectious agents is to know the gut microbial diversity of this bat species. So the Tenochirus jaegori, which is an endemic fruit bat to the Philippines, presents a unique subject as a source, as a study subject to determine the microbial diversity and the taxonomic profile of the gut microbiome, hence the objective of this presented paper. So for this study, we collected samples from the vicinity of the Cathedral Cave in Cavinti, Laguna. Prior to sample collection, we have secured a wildlife gratuitous permit from DNR, and our gut collection from the collected bats was approved by the UPLB Institutional Animal Care and Use Committee. So the bats were caught using mist nets at the early evening while these bats are out to hunt for food. So the collected bats were identified by the Zoological and Wildlife Museum of the Museum of Natural History. And because of the nature of the study, the collected bats have to be sacrificed. So immediately after euthanization, the guts were collected and they were placed in 95% ethanol and were stored in liquid nitrogen and then transported into the laboratory. Now, because of the diverse functional activity of the gut, the whole intestine was used as a sample for collection. So we opened the intestine and we scraped the gut mucosal lining tissue as well as the gut contents. And then a differential centrifugation was done to separate large particulate matter from the microbial cells, and then these were pelleted, and then when the pellets containing the microbial cells were used for total DNA isolation. After quality control, the enough concentrations of total DNA was sent for sequencing using Illumina MySeq platform with 300 BP per run setting and a throughput of 100,000 reads per sample. For the bioinformatics analysis, the qualitative or the quantitative insight into microbial ecology version 2 or the time 2 software was used as the main analysis tool. So first, of course, we imported the raw sequences into the software, and then we used Zymos Figaro to determine the optimal truncation parameters objectively prior to quality control filtering. And then DADA2 plug-in within QIIME2 was used to QC filter the raw sequences, denoise the raw sequences, and then merge the paired end reads, the forward and the reverse reads. After which, we also removed the chimera, and then a feature table containing the amplicon sequence variants was outputted by this plugin. The Silva 138.1 database was used to train a feature classifier, and this feature classifier was used as to assign taxonomy to the valid sequences. So this table shows the sequence statistics of the samples. So the total raw sequences was 559,663, and after filtering, denoising, merging of paired end reads, removal of the chimeras, and then filtering of non-bacterial sequences, the final valid reads came to about 441,721 reads, and these were assigned to 183 amplicon sequence variants. The 183 amplicon sequence variants was assigned to 11 bacterial phyla, 11 shared bacterial phyla among the 5 samples. So we've collected 5 individual bats. So as you can see, apparent abundance was observed from Campylobacterota, phylum Firmicutes, and phylum Proteobacteria at 51.33%, 38.82%, and 9.47% respectively. Okay. As you can see in the table as well, 73 shared genera was observed among the amplicon sequence variants. So this was observed were Mycoplasma at 35.99%, Helicobacter at 51.36%, and Urea plasma at 1.36%. As you can see in the table, other potentially pathogenic genera of microorganisms were also observed, such as Urea plasma, Klebsiella bacillus, and Clostridium sensu stricto 1, as well as members of the family of Enterobacteriaceae and Neisseria. So the result of our study of this paper was corroborated by other previously published microbiome studies of the gut microbiome. So Banskar and his colleagues in 2016 reported a high levels of OTUs of the Mycoplasma, which was observed in 3 individuals of Cynopterus species. Cynopterus is also a fruit bat. Another study by Hatta and colleagues in 2016 reported a 5.1% relative abundance of the family Enterobacteriaceae, from 67 pooled samples of Rosetus amplexicaudatus rectal swabs, which was collected here in the Philippines. Lastly, a study by Lee et al. Compared the bacterial gut microbial diversity of bats based on their diet. So they compared the gut microbial diversity between insectivorous bats and fruit bats, and they found out that there's a significant difference between the microbial diversity between these two types. In particular, they were able to found out that the families Mycoplasmataceae and Clostridiaceae, as well as the genera Ureaplasma and Klebsiella, were also observed in fruit bats as compared to insectivorous bats. So these previous studies corroborated with the results that we were able to obtain from the microbiome analysis of the samples that we've collected from the Cavinti, Laguna. Okay. So the presence of pathogenic or potentially pathogenic microorganisms were determined from the gut microbiome of the Philippine endemic fruit bat, Tenochirus jaeguri. And to our knowledge, this is a first report of the gut microbiome of this bat species. And the results of this study reveal insights into the gut microbiota of Tenochirus jaeguri, its potential as a source of zoonotic pathogens, and it highlights the need to know the role of these groups in bat health and therefore and for their control as emerging zoonoses. While we have established the gut microbiome of the Philippine endemic fruit bat, this is just an essential first step. And we still would like to know the role of these different genera of microorganisms in the guts of this Philippine endemic fruit bat. So right now, we are currently doing a functional profiling based on the 16S metagenomic sequences we obtained from these samples. There was also a relatively high abundance based on the presented table, abundance of the genus Helicobacter in the gut microbiome of Tenochirus jaeguri. And this is something that we would also like to explain more, or at least to learn why there is such a relatively high abundance. So this is currently a continuing project, and with the number of samples presented here, we may not be able to glean an insight on why there is such high abundance. But we do have other sampling sites, and we were able to collect other bat species of other individuals of the Tenochirus jaeguri species. And we would also still, again, like to determine the microbiome of those and compare and see if there is a similar trend with, of course, the Helicobacter and other microbiome profile that we've already collected. Aside from Tenochirus jaeguri, we also have other bats collected from these different sites, not necessarily endemic to the Philippines, but the results would provide us an insight into the microbiome profile of different bats that we found in Calabarzon, and this may provide an insight, and it can give us knowledge on possible pathogenic microorganisms, potentially pathogenic microorganisms that maybe are re-emerging or emerging zoonoses. So these are the references used on this paper. And I would also like to acknowledge the following agencies: the OST, the NICER program, the S4CP, the OSTP card, the municipality of Cavinti, Laguna, and its Menro, the Institute of Biological Sciences, and the Microbiology Division of UPLB, where some parts of this study was conducted. Thank you.
Thank you very much, Mr. Datul. We have final questions for our final presenter. We're starting with our chair, Dr. Cabrera. Go ahead, Po.
I give you the honor.
Wow, are you sure, ma'am?
Okay, Dr.
I might
Reyes po.
monopolize. Okay, wow, finally it's good to put a face to the name because I only get emails from Bonnie Datul. Now I know how Bonnie looks like. So nice to meet you, Bonnie. My question is on, first, actually, I have, if will be allowed, at least 3 questions. But you said that you sampled the entire gut for metagenomic DNA extraction. Can you just describe to us, because I don't think that was operationally described in your manuscript, how did you, what was your sample processing design to ensure that you got a representative sample of that entire gut that would represent would give you good coverage of the microbiome in that entire gut?
Okay.
So that's my first question.
Okay. Thank you, Dr. Alios. So my apologies if it wasn't specifically mentioned in the manuscript. So we followed the method of Phillips et al. On a published paper in 2012. So we excluded the stomach. So when we collected the bat gut from the bat itself, we excluded the stomach. So we just from the end of the stomach, the start of the large intestine up to the anus. So that is the length of the intestine that we've collected, and then the entirety of this the intestine was as described by Phillips et al. We opened it longitudinally, and then we scrape off the mucosal tissue lining as well as the gut contents. So that includes leftover foods currently being digested by the intestine. So we collected all the gut contents as well as the mucosal tissue lining, and then we suspended that in sodium chloride in saline solution with twin 80. So that was based on a protocol by Nordgaard et al., which was published in 2005, wherein because we scrape the mucosal tissue lining, what we've collected are not only the microbial cells, but also the host cells, as well as some food particles. So to exclude those during the total DNA extraction, we did the low-speed centrifugation, and that is to separate the large particulate matter, which may include the food particles as well as some host cells, and left with the supernatant would be the microbial cells. So we collected the supernatants, and then we pulled the supernatants, pelleted it, and then the pellets was used for the total DNA extraction.
So how many grams would the pellet be? And do you use the entire pellet for DNA extraction?
So yes, Dr. Al, we use the entire pellet for the DNA extraction. So we were unable to measure the weight of the particular pellet prior to DNA extraction.
But that you believe is representing the entire microbiome from stomach to anus, right?
I believe so.
So I guess because it's a technical paper, I think gut is not a technical term. Is that, I don't know. But maybe it's better to really indicate the, what do you call that? Yes, the sample. Because when you say gut, it's my gut. Yeah, it's laymanized. Yes, thank you, Dr. Cabrera. It's laymanized, yes. So just for future, maybe if you have opportunity to improve the paper, is to use the actual parts, the technical names of the parts. Okay. Anyway, my second question is, you mentioned you talked about core microbiome, right? That you said that the 5 individuals, bat individuals, showed 73 shared genes, and this is the core microbiome. What is the technical definition of a core microbiome? And would shared genera across 5 individuals, would that qualify as core microbiome?
Yes, thank you, Dr. Alios. So I guess when we say, excuse me, I guess when we say core microbiome, it is something that can be found among different individuals or among different samples from the same species, like in this case, from Tenochirus jaeguri. So I would say that perhaps 73, when I say shared genera, so it is the totality among 5 samples, but not necessarily that all of each of those 75 can be found in each individual bat. So I guess with the term core microbiome may have been not appropriate when I use it with this presentation. But thank you for that enlightening.
Okay po, we have to move on po with another judge as we are very pressed for time.
Ubusin ko na
Dr.
raw.
Dalisay po.
Just say, can I just comment on that? Yeah.
Very
Because
quickly.
when we talk about core, hindi po daw ako makatulog. When we talk about core microbiome, I think functional metabolism, functionality is part of the definition. So just having, maybe you're right, maybe the better term is just shared genera and not necessarily core. You need more, analysis to really say this is core microbiome.
Thank you. Thank you, Dr.
Thank you,
Alias.
Bonnie. No, I give my right to my esteemed colleagues.
Sige
Thank
po.
you, Dr.
Salamat po.
Cabrera. So, Bonnie, so first of all, I would like to commend you for working on something that is difficult because one is your sampling. Second, capturing this mammals is also very difficult. And third, getting the microbiome in the gut is also difficult. So not everyone can do it. It's a very good opportunity for you. So I commend you on working on that.
Thank you, Dr.
Okay.
Dalisay.
First question. You presented a microbiome list, and I was wondering if I haven't seen any viruses on your list. Was there any?
I think that is. Excuse me. We were only trying to identify the bacterial bacterial microbiome. So we were unable to detect, I guess, viral. Sorry, did I heard correctly? My apologies.
Yeah.
About viruses. So we used for the microbiome analysis. We use a specific gene for bacteria and archaea, for prokaryotes. So we targeted the variable region V3, V4 of the 16S rRNA gene. And hence, during the analysis as well, we excluded non-bacterial sequences.
Okay.
And therefore, the results after the analysis only outputted bacterial genera.
Is it something that you are looking into in the future? Like, what are the viruses present in these samples?
I'm not sure if I can share this, but it is definitely one of the offshoot projects that we are looking for from this program, is to determine the virome of bats. Because technically, COVID-19 was caused by a virus. And as I think I've mentioned during the introduction, that the bats are well-known reservoirs of viral and bacterial pathogens. But for this paper, we are only able to present at this moment the bacterial microbiome profile of this particular species of bat.
Alright. Okay po. Sige
Yes,
po.
thank you.
Thank you very much. And a round of
No,
applause,
comment
please.
only.
Ay, okay, sige po.
No question.
One
comment.
final, ma'am, please.
No, I was going to say, because of you made use of the 16S rDNA primers, right?
Yes, ma'am.
So I suggest that you make your title more specific, probably bacterial microbiome, because you didn't look for fungi either. You didn't use 18S, for example, or your ITS. So just make it more accurate.
Okay. Thank you, ma'am, for that suggestion.
Thank you. All right. A round of applause, please, for Mr. Datul. Congratulations, Mr. Datul. All right. The presentations were very thought-provoking and timely. As much as we would want to extend our discussions, it is time for us to end the competition. Once again, we would like to thank all our finalists for for presenting their works during this PSM 50-second Best Paper competition. They passed through 2 stringent screening processes. So congratulations na to all of you. We know that our participants' curiosities, both online and on-site, were met during this session. So before we formally close our session today, may we call on the chair of the Best Paper Competition Committee, Ms. Hannah Jane Dean, and PSM President Dr. Howell Haw, to award the certificates of appreciation to our esteemed judges. We would like to also call on stage already our judges, Dr. Ralios, Dr. Delisay, and our chair, Dr. Cabrera. While they are coming on stage, allow me to already read the citation. The certificate of appreciation is awarded to Dr. Ralios, Dr. Delisay, and Dr. Cabrera for having served as judge in the Best Paper Competition professional category during the 52nd Annual Convention and Scientific Meeting of the Philippine Society for Microbiology, Incorporated in cooperation with Trinity University of Asia, with the theme Microbiology for Unity and Prosperity through collaborations and industry linkages, held at the Hilton Manila via hybrid platform from July 20 to 21, 2023. Given this 20th day of July 2023, signed Hannah Jane Dean, the chair of the Best Paper Committee, and Howell Haw, PSM President. Thank you very much to our judges. Reminder to everyone that the awarding ceremonies will be held tomorrow, July 21, 2023, Friday, 4 o'clock p.m. onwards. We again thank everyone for making the PSM 52nd Annual Convention and Scientific Meeting Best Paper Competition Professional Category a success. Once again, this is Pierangela Vital, the Division Representative for Aquatic Microbiology, your moderator for this session. Thank you and see you all tomorrow.
This event would not be possible without the support of our sponsors and exhibitors of this year's convention. We'll be sharing an audiovisual presentation prepared by Diamed, Yana Commodities, and TNC Everlight. An open forum would follow the presentations.
Science, one word that encompasses so many things, people and life in general. From the materials of the house we live in to the safety of the food we eat, science builds the foundation of our daily life. Yana Commodities Incorporated works behind the scenes, helping scientists create, develop, and improve our lives. Benefiting society is not just a social responsibility, but also the mission of our company. Opening its doors in 1979 as a small company selling laboratory supplies, Yana Commodities has grown considerably. Supplying over 5,000 products to 1,000 active customers and 100 dealers all over the Philippines, Yana now employs over 70 experienced staff dedicated in assisting our valued customers. It is the mission of the company as a distributor of laboratory equipment, chemicals, and supplies to benefit society through 4 aspects. Product availability. Yana keeps a high level of inventory so that when you need it, we can serve it. Reasonable prices. Here in Yana, we do not promise the lowest prices, but we will always give a fair and reasonable price. Fast service, be it delivery or technical service, you can depend on us. Our experienced service technicians will surely do their best to repair, test, and even calibrate the unit. And lastly, education. Yana holds free monthly seminars in order to increase market awareness, because with knowledge,
each and every one of us can better serve our society. We in Yana take our social responsibility very seriously. Annual distribution of goods to the less fortunate with a Christmas party gathering for the kids that is both educational and fun. Yana serves a wide range of clients from the industrial, government, life science, education, pharmaceutical markets spanning across agriculture, mining, food and beverage, dairy, hospitals, power plants, petroleum, all the way to research and development. Yana Kimartis believes in serving the best to our customers and has kept their facilities in good quality condition. Our facilities include a specific control temperature room for the culture media, a cold storage room for preserving cold temperature products, ASTM water production room, display room, and our very own seminar area where we hold our free monthly seminars. With over 5,000 products on hand, Yana strives to be the one-stop lab shop throughout the country.
Good afternoon, everyone. As we listen to the different product presentations this afternoon, I hope everybody will also enjoy their afternoon snacks. So earlier, we have seen the video of Yana Commodities. Should you have any questions regarding their products, you may visit their booth outside of this hall. To continue, we will now proceed with our first live product demonstration or product presentation from one of our sponsors. Okay, I would like to call later after the product demonstration our business manager, Dr. Rodilio Flores, to moderate the open forum. So let me introduce our speaker. Okay, he is currently the business development manager of Copan, the partner of Diomed, one of our sponsors for this annual convention. Let me call on stage Mr. Arcanado Danay. Sir.
Thank you. Good afternoon, everyone. Today, I would like to talk about the HPV, and especially I will focus on the self-sampling. HPV is one of the main causes of the cervical cancer, but paradoxically, it is one of the easiest to treat if it is detected in the first stages. So, but in order to detect as much cases as possible, we need to increase in each country, we need to increase the screening programs, so the national screening programs. And how can we increase the national screening programs? Well, there are different ways to increase it. First, to invest more in the healthcare, or on the other case, to find a way to increase the screening programs by making it easy, cost-effective, and accessible to everyone. Actually, the last decades have seen a progressive transition from the traditional pap test to the HPV DNA testing, and there have been several reasons why. The last one has been the catalyst has been the COVID pandemic. Why so? Because all of us, we know how it affected the healthcare sector, both from the professional side, so the healthcare worker, but also the access to the healthcare facilities. This is why in 2021, the World Health Organization decided to endorse and to promote the self-sampling as a correct method, as an alternative method to the to the practitioner collectioner method. We in Copan have a long history in microbiology, especially in the pre-analytics. So we decided, we have been working in this field since 1979, and we decided to exploit, so to use our knowledge, our experience, to find a way to make a device that could help or would increase the participation, the attendance of the people on those screening problems. This is why 6 years ago we launched in the market the self-vaginal flock swab, which is a dry swab, and it is dedicated to the collection of vaginal sampling that will be tested for viruses, fungi, bacteria, and HPV for sure. Now, I would like to draw your attention about some of the advantages of first the self-collection and also to our device, which is the flock swab. First of all, we use the flock technology, which is basically a complete different way of doing a swab. So we have, in the picture here, we can see two swabs. We have the traditional cotton swab and the flock swab. There is a huge difference between one and another. So on the left, we have the fiber swab, and we see that the structure is a chaotic one. What will happen with this chaotic structure is that we will have the sample, but we will not be able to release the correct amount of sampling. On the other side, we have a more regular structure, so the flock swab, which uses nylon fiber, which is an opten structure, and it will release from 80% to 90% more than the traditional one. Now, there is a short video that I would like to show you, if it starts. Okay. Can I just, I'm trying to, I'm trying to share the screen. Sorry? No, I just want to share the screen because it's not sharing. Can you just... It's not sharing anymore, so maybe if you... I wanted to share the video, but it's not sharing anymore, so can you maybe just...
Sir, you just need to play it.
Yeah, I mean, to show it there.
Sir, pag pinlay, sir, lalabas po. Just play, sir.
But they cannot see it.
I will just plug it, sir.
You will... All right, yeah.
Sir. Playing na, sir.
Yeah.
Audio, sir.
It's okay. Yeah, all right. We can just skip it. It's fine. So how we go back to the... No, it's fine. It's fine. Well, it seems that no way to make this video working. Anyway, so I will just use the pictures here to show you that actually we see that the structure of the flock swab is completely open, and as I said before, it will release up to 90% more sample. So using the nylon instead of the cotton swab, we'll have at the end a soft and smaller swab that will be painless and can be, let's say, more comfortable for the women. This is a study I just wanted to share with you. It has been a comparison between the brush-based sample collection and the flock swab. I will just directly go to the conclusion. Those are online studies. So the women that has been tested with both devices, they preferred the method that used the dry swab. And the larger number of women reported feeling very confident in using compared to the brush-based devices. And the reason why they preferred is also because basically in 6 steps, it is an easy-to-use device, and in 6 steps, very easy to understand, you perform the collection. So within 30 seconds, the women will collect the correct amount of the cells to be tested for. This is another study, and the results are that 91.5% of women found the dry and flock swab very easy to use, and around 90% confident in correctly using the device. All right, okay. How about the transport and the performances? It is, as I said, it's a dry swab, so it will come in a dry tube, and related to the performances, after the collection for the HPV testing, it is stable at room temperature, so between -20 to up to 50 degrees, it will be stable for 2 weeks. However, there are some independent study, the one that we can see here, made from a diagnostic company. They tested the device, and it is stable for 30 days, for up to 30 days, for 30 degrees. Thanks to those tests, we have validated the device for different platforms, and different platforms has been evaluated using the flock swab. It has been validated up to now from the BD on Clarity, which is actually together with Roche Cobas, one of the main used for the HPV testing. However, there are other performance evaluation done with different brands and different platforms. Now, in the next slides, I would like to share some studies that has been conducted, some data. Some of the most important, we have 3 here, has been conducted in 2 in Europe, in Italy precisely, and 1 in Australia. So we have European values and extended values. They both validated the cell sampling, and in the case of extended values, it validated the BD core diagnostic platforms, and which is basically you can find it in the most of the laboratories. Another, more interesting is Scope 1. It has been conducted in Australia, and it validated and evaluated the flock swab device with all those companies, all those platforms. So BD, Abbott, Roche, Cobas, and each of those studies actually has been quite successful, and the attendance of the women to those programs quite high. Now, this is just an example of one of those studies. Here, 744 women were enrolled, and for each woman, 2 samples has been collected. One, which is the clinical collected sample, and the other one, the self-collection, which has been done in point of care. So the conclusion are there. Our study, so the study shows that over 90% of correlation between the self-collection device and the clinician collected, and the cervical sampling. This is why all those studies actually resulted in an increase of number of programs, national programs that utilize the self-collection device. This is just a map of some of the main the most important studies that has been conducted up to now. So you can see more or less in United States, more or less in America, it has been in North America, everywhere. A lot of countries started to use it in Europe and in Australia as well. So this is the first established screening program, national program in Australia. And they started to use the self-collection device 2 years ago. One which I think is way more important for us since it is, it is near Philippines, so in Southeast Asia. This is the program, the screening program of the Ministry of Health of Malaysia. So they started this study, this screening program, a pilot program in 2020, and they extended from 2021 to 2024. Up to now, I had the pleasure also to meet some of the people in the Ministry of Health. Up to now, this has been considered that Malaysia is one of the majority of the women are, let's say, Muslim. And this has been, let's say, a game changer since it enrolled more women. Instead of going to the gynecologist, for cultural reason, is not always possible, they prefer to use the self-collection device, which they can use it at home or in the point of care of the clinic. Another interesting study from interesting program from Malaysia is the Rose Foundation one. So basically, Rose Foundation, it is a no-profit organization, and they are engaged in increasing the number of women enrolled for those programs. And they're traveling all Malaysia, so the most remote areas, and the result that they've been conducted first, the number of women that attended those programs has been extremely high compared to the past, and also the performances that has been shown with the self-collection device are comparable to those collected by the clinical. Now, this is the last study I wanted to mention. This has been conducted in Western Pacific Islands, and the reason why I mentioned this study is because it shows it was, let's say, conducted in extreme temperature compared to, let's say, Europe or United States. So temperature which are similar or even higher than Philippines. And this has been conducted and showed the good performances of the self-collection device through the flock swab for up to 2 weeks. Now, last but not least, we are also working here in Philippines together with Diamed and one university in Manila. We're working about for a program, national program, and the self-collection device has been introduced as alternative to the clinician collection sample. The first studies, we had the pleasure to meet yesterday one of the people that are involved, and the first result that we had from locals, so from Manila, has been quite interesting, and for sure the performances are great, and also the number of women that were enrolled, let's say, seems that based on the interviews they had, seems that they feel more comfortable to do the self-collection at home instead of going maybe, I mean, you also here in Philippines has also the jam problem, the traffic jam, so they prefer it to do at home instead of going maybe to the laboratory or to the clinic or to the hospital. So to conclude, what studies showed related to the self-collection is that it can be the best solution to try to increase the number of attendance to those screening programs and to fight the HPV. For sure, we can have the best, let's say, the easiest or the most comfortable or cost-effective devices to increase the number of women that will test themselves. However, everything should, let's say, we everywhere, not only in Philippines, everywhere, I think that we should invest more in healthcare, especially for those situations when basically a test can save lives. Because for sure, HPV can be defeated. And we for sure, in Copan, we will, let's say, engage. We'll continue, do our best to find a way to fight it. Thank you very much.
Again, a warm round of applause for our product presenter for this afternoon, Mr. Arcanado Danay. Thank you very much for sharing the wonderful work and products of Copan in terms of HPV collection. Do we have questions from the audience? I know you are very eager to know about HPV. Yes, Dr. Penular.
Thank you very much, sir, for the presentation. So if the ultimate goal is to reduce cervical cancer due to transmission of HPV of cases, so you've discussed mainly the kit for women, but of course we all know that men can also carry HPV, although they are mostly asymptomatic, but they can still transmit. But do you have an equivalent test kit for males? Thank you.
Thank you, doctor, for the question. Look, it's an interesting question because we are working on it. We are working on it. We will, now, it is still, we will launch a new product, which will be in September. Our idea is to use urine, so to detect STIs and HPV through urine. And definitely, this will be dedicated also to men. Now, I cannot share, since it is still an ongoing project, I cannot share much information and or some, like, we are doing some studies, some evaluation. I think it's still too early from my side to share this. But it is, I mean, thank you for the question because we are working on it. We have been working actually in the last 2 years about it, but for some regulation, we decided to postpone the launch of this new product. But yes, urine will be our next target for HPV and for STIs as an overall. I hope that the next time I will be able eventually to present to talk about the urine collection. Thank you, sir.
Thank you very much, Dr. Pinular. Do we have more questions? Last one question for this round. None? Again, the warmest round of applause to Mr. Arkinado Danay of Diamed Enterprises. They will be with us until tomorrow. Now, their booth is located in the hallway, so please do make it a point to visit their booth and ask more questions about their products. Thank you very much, everyone.
Thank you, Mr. Danay and Sir O'Neill. Now we proceed to the next live product presentation. May we invite Meng Hua Ong, the current applications and business development specialist from the Bruker Microbiology and Infection Diagnostics, partner of TNC Everlight, also one of our sponsors for this annual convention.
Test. Okay, good afternoon to all the honored guests. I'm very delighted to be here. Today I will be actually giving a presentation that is related to the microbiology solutions. So this microbiology solutions is going to cover the research, the food, and also the industry market. So my name is Meng Hua. I come from the Bruker, the partners of TNC Everlight in Philippines. So what I'm going to present is actually an equipment that is called the multi-biotyper that can be used in doing microorganisms identifications. So the applications for microorganism identifications can be that you are doing a quality control of your ingredient to check your end product whether it's free from any of the contaminants, to detect any pathogens that is present in your product, regardless it's a food, it's a product, skin products, pharma products, or drugs. And of course, we also want to monitor the hygiene and also your water in the production sites to ensure that all these are clean and we don't have any food spoilers or any spoilers into your end products. So the principles of identifications is actually very straightforward. So any of the microorganisms that grow on your non-selective or selective agar, we use the colonies, we prepare it, and put into the equipment, the multi-TOF systems, and acquire the proteomics fingerprints of the organisms. And later on, based on this fingerprint of the organism, because each of them has their own unique fingerprint, just like our human fingerprint, we will be able to know what is the identity of the microorganism. And the whole process of the identifications from sample preparations until you get the results for 96 samples will only happen in 30 minutes. So this is a very easy, faster than any of the convention methods. So if any of you are already doing microorganism, years ago we are actually doing biochemical tests that you need at least 1 day to know the identity of the microorganisms. But with this technology, you will be able to get the results within minutes from the culture plate. So the technology inside is actually based on a multi-TOF technology. So the full name of multi-TOF is matrix-assisted laser desorption, ionization time of flight. So basically, we prepare the samples, which is the colonies, in together with the matrix solutions, put into the equipment. The equipment will activate the proteins present in the organism, and then we will capture the proteins using the detector unit on top of the equipment. And when we get the proteomics fingerprint of the microorganism, it is very important that we have a good database or a library that we can use it to find the identity of the microorganism. So in Bruker, we actually have a very big database covering 4,274 species of the microorganism. And if you look into our development of the database, we actually update our database on a yearly basis, and we start off with 1,850 species, and we have now in 2022 the latest update with 4,274. So the running cost is actually going to be much more lower than your conventional biochemical test because we don't need to use a lot of reagent. There's only three, the matrix solution, the QC material, and also the plate for you to prepare the sample. So ideally over here that you are going to save your time and also going to save your running costs. And we do not need the lab to also first know whether this organism is a gram-negative or gram-positive organism to choose the correct consumables to use. So these same consumables is applied to all types of microorganisms. So if we want to look into how a multi-biotyper can really fit into any of the identifications methods, so over here is the ISO 6579 showing for the Salmonella detection workflow. So by using the routine method, you will need 5 days to finish your workflow for the confirmation of the pathogen. But if you use a multi-biotyper to replace the biochemical test, you will have your workflow completed at day 3, which able to help you to save 2 days. So our multi-biotyper technique to do the identifications for the foodborne pathogen is also certified by Microval for the ISO 16140 Part 6. So there is 4 certificates that was issued from Microval, and this is currently valid until 2026. So every 4 years, they will do a renewal. On top of that, our method is also mentioned in the AOAC official method of analysis. There is 2 chapters within it, and also is tested by USDA Food Safety and Inspection Services. So this is all both mostly related to the food industry, food safety portion of food regulation. In terms of the compliance part, which is more relevant in a pharma settings, so our software actually comes with a compliance assistance module that is compliant to 21 CFR Part 11 that includes the audit trail, the authentications, data integrity, and also signatures. So in terms of our database do also covers a wide range of the environmental samples and also samples that is present in animals. So this is also very good for us to do environmental monitoring and also animal health. So in short, the benefits of a multi-biotyper is really a very fast identification. You save a lot of time. Imagine if you have your product that is going to release, but because you are just waiting for this result for 1 day, 2 days, this will actually cost you a lot of money to keep your products in the warehouse. And it's a very reliable technique because we do actually have a lot of validations from different entities to show the performance of the equipment to correctly identify the microorganism. And next, I will move on to another equipment that is if you need some things that can do a strain discriminations. So just now was identifications that was up to a species level. But after you know about the species, but you want to know in depth more about the organism, so up to a strain level. So we have IR biotyper, which is equipment next to the multi-biotyper that was shown in the picture, that this is actually able to help you to give you more information on the strain level data. So IR biotyper is actually based on another principle, which is FTIR technology, Fourier transform infrared principles. So what it does is actually that it is capturing the cell surface fingerprint. Just now, the multi-biotyper is the protein fingerprint, but IR biotyper is capturing the cell surface fingerprint. So with this, we will be able to know more information of the microorganisms at a strain level. So this is actually good that if you are going to identify a source of a contamination. So for example, in a lab, any type of a lab, you found a contamination of an E. coli, but you would need to know where is the source of contamination so that you can decontaminate the surface or the equipment or the lab of the source of the contaminations. So by using the IR biotyper, you will be able to find the source of the contaminations within 6 hours so that you can start to already do the decontaminations, and you don't need to decontaminate your whole facilities just because you found a contamination. So this is one of the studies that was done by Food Safety Net Services, where they actually helped to find the source of E. coli contaminations in slaughtering facilities. The second portion is, I believe that most of the guests over here, you guys are also looking into probiotics studies because this is some things that a lot of countries are focused on because of the benefits of really having these probiotics. And probiotics, when we want to do some kind of a production or put them into an active ingredient, just to know that there are just to know their species is actually not enough and not sufficient. So with the IR biotyper, you will also be able to get more information of the strain level identification of the probiotics organisms. So in short, the benefits of IR biotyper is that you can get rapid, fast strain discriminations results. And it's a very small equipment and it's cheaper compared to doing a WGS. So this can also be your in-house solutions, especially if you are a private industry or companies that is producing probiotics. You do not want other people to know what strain you are using it in your product. So this will actually be a very good solution that you can keep everything in-house. You don't need to outsource it to other people. So same goes with contaminations. So when a contamination actually happens in your lab, you also do not want other people to know that there is a contamination happen. Yeah, so this is also going to be a very good equipment that you can have in your own lab to do all the hygiene monitoring and also the R&D work for your products. So in summary, from Brookers, we have the multi-biotypers that is able to give you the species identifications of the organisms where you actually can use it to do monitoring of the pathogen detections and also to know what are the microorganisms that you are using it for your food production, for examples. And of course, on top of that, you can also use the IR biotyper together for a strain discrimination and also a hygiene questions that is happening in the microorganisms or QC lab. So with that, this is just showing you the current install base of our multi-biotyper in the globe. So at the end of 2022, we already have a 5,700 multi-biotyper system. This also does include the clinical, the hospitals lab as well. And if you do have more questions that you would like to reach out, we are actually at the exhibition booth. Our local partners here is TNC Everlight in partnership with Becton Dickinson. Okay, thank you.
Thank you very much, Mr. Ming Hua Ong. Do we have questions from the floor? I know most of us are handling pathogens in our labs. So would you like to acquire probably one multi-biotyper? You may want to ask how much one unit costs. No questions? Let me ask about the fidelity or the veracity of identification, since this is based on biomolecules, perhaps. So, but when you check the accuracy of identification, does it fare with the conventional or gold standard identification of pathogens?
Yeah, so the identifications that the multi-biotyper is doing is that we acquire the proteins profile of the microorganism. So it's a little bit different with a biochemical test, but of course we do have studies that we have already compared the accuracy of doing the identifications with a multi-biotyper using a biochemical test and also a 16S sequencing method as well. So actually our method is actually more reliable as compared to the biochemical test, and it's almost the same sensitivity as the 16S sequencing method. This is because actually there's a lot of closely related microorganisms that actually share the same biochemical results. So at certain points, some of the microorganisms, you are unable to actually differentiate using a biochemical test, but of course with the multi-biotyper, it will give you a better accuracy of the identifications.
But it calls for culturing the microorganism in a specific sets of conditions.
No, there is no specific conditions. So that's why in just now one of my slides I did mention that you can use the organisms that is from your selective agar or a non-selective agar. So as long as the colony is grow on your agar plates, then these organisms can be used to do our identifications. There is no limitations in culture conditions.
Well, that's amazing. Any more questions? I do have tons of questions, but I would rather go to your booth and maybe engage in a very good conversations, and maybe a lot of you can join me in, going to their booth and probably getting some of the freebies that they have. Thank you very much.
Thank you.
Mr. Menghua Ong from TNC Everlight.
Thank you very much. Now we shall hear once again from Diamed Enterprise. This is actually part of their previous presentation. However, we had some difficulty earlier. So just to complete their presentation, let me call Ms. Janela Arganda, the Technical Sales Representative from Omics of Diamed Enterprise.
Good afternoon, everyone. My name is Janela Amarganda, and I am the Technical Sales Representative from the Omics segment of Diamed Enterprise. So community and accessibility. These past few years have shown us that there is a demand on genetic sequencing, and the field of microbiology is no different in this regard. And now, more than ever, calls for the accessibility of these technologies and our unity as a scientific community. So once again, everybody, I am Janela Arganda. Today, I will be presenting Oxford Nanopore Technologies. So Oxford Nanopore Technologies has been around since 2005 and is a biotech company based in the United Kingdom. And ever since, their goal has always been to make the analysis of anything by anyone, anywhere. These are just some of the institutions that are currently or have used Oxford Nanopore Technologies as their sequencing technology. So the heart of the nanopore technology lies in its very name, the nanopore. So it is a biosynthetic engineered protein molecule, and the sequence is determined by disruptions in the current detected by the nanopore, resulting in electric signals that are then interpreted as base call data. Now, this technology sets ONT apart in various ways. First of all, Oxford Nanopore Technologies boasts its ability to read ultra-long sentences, but it does not shy away from its ability to read short ones. In fact, it could read from sequences as short as 20 bases and as ultra-long as 4 megabases, with a speed of about 420 bases per second. Real-time data analysis is made possible with immediate actionable results, and workflows can be customized. Oh, I'm so sorry. Now, the sequencing yield is scalable to fit your needs up to 14 terabases. Now we will see later the variety of devices and how from a small MinION device could be scaled to the bigger, higher throughput devices. Next, the workflows can be customized according to your application and with prep time that can be as short as 10 minutes. Now, the portability of this device allows for the use on versatile settings with no need for major facility refurbishing. Now, Oxford Nanopore Technologies has multiple purchasing options which can assist researchers with limited budget. Now, Oxford Nanopore Technologies can be used in a multitude of applications, and one of such as whole genome sequencing, targeted sequencing, RNA sequencing, metagenomics, epigenetics, and shotgun sequencing, and other techniques applicable for microbiology studies. Now, these applications, along with the appropriate kits that would be based on your workflow, are then sequenced by a line of powerful sequencers, such as the MunION, which can be seen, an example could be seen, which is the Mark 1C in our booth, which you can visit later. This particular line of sequencers can have a theoretical max output of about 50 gigabases. The GridION, which has a theoretical max output of about 250 gigabases, and the PromethION, which could have a theoretical max output of about 14 terabases. Now, the data analysis is then performed on analysis tools as with a device such as the device integrated Minnow and the Epitome cloud-based analysis platform where post-sequence analysis is done. And as I introduce Oxford Nanopore Technology to the Philippine Society of Microbiology, we're pleased to announce that as of last week, Diomed Enterprise is now the official distributor of Oxford Nanopore Technologies in the Philippines. In line with this, now is the time for us to join the genomic revolution and make this technological progress accessible to us all. Thank you very much, and please visit our booth for further details. Good afternoon.
Thank you very much, Ms. Janela. Again, please do visit their booth. They are the official distributors of Oxford Nanopore in the Philippines. Hopefully, all our labs will have this Oxford, para we can do more good research in the Philippines. Maraming salamat. Thank you very much, Ms. Janela.
Thank you din po.
Please do visit their booths tomorrow or after this session. Thank you very, very much. Ms.
Thank you, ma'am. Thank you very much, Sir O'Neill. We're almost at the end of our day one for the 52nd Philippine Society for Microbiology Annual Convention and Scientific Symposium. But don't worry, we still have a lot of activities that we will do for the rest of the day. I would just like to announce that our next activity is the General Assembly and Business Meeting for all members of the Philippine Society for Microbiology. The business meeting and General Assembly will be in this hall, Hall A. So may we request for all members to please stay in Hall A for our General Assembly and Business Meeting. To our other participants, we would like to invite you to view all the different poster presentations in our ballroom hall, and you can also visit the exhibits of our sponsors. But I also would like to invite everybody for our fellowship night tonight, which will be conducted via online. I believe our secretariat has also emailed the Zoom link for our online participants. So see you tonight for our fellowship night, 8 p.m. po tayo, via Zoom, and for our on-site, of course, dito sa Hilton, Manila. Probably, I guess, in 10-15 minutes, we'll start the business meeting and General Assembly. Have a nice day. Malapit yung place na po ito to those traveling or who came all the way from Mindanao, Visayas, Luzon. There are a lot of pasyalan po, mall, casinos, shopping area. This place is full of shopping places. So I hope you'll enjoy your stay in Manila. Thank you. Hello everyone, just a reminder, the registration for tomorrow will start at 7 a.m. Thank you. Good afternoon, everyone. May we call the following members of PSM? Okay, if you are here, can we request you to please approach Miss Papa, Dr. Papa, okay, on that side. Okay.
Hello.
Dr. Sheila De Jesus,
Naku na po, huwag yung pipindutin. Wala na po pipindutin.
Dr. Susan Sedano,
Kailangan nyo pa isa.
Dr. Charina Grace Banaay, Dr. Cynthia Lim Saiko, Dr. Maria Teresa Perez, Dr. Resurrection Sadaba, Dr. Adrian Marie Hanagap, Dr. Malem Flores, Dr. Remedios Flamiano, Dr. JC Pagirigan, Dr. Marie Christine Obusan, Dr. Rohani Navarro, Dr. Michael Angelo Nicdao, Dr. Fatima Cruzada, Dr. Walter Lavinia. Thank you once again, the names that were called. If you're here, kindly approach Dr. Donna May Papa. Nandun po siya. Thank you.
The microbial world, so fascinating and diverse. In the last 4 decades, the study of microorganisms has grown by leaps and bounds. Scientific advances have led to our understanding of the microbe's life processes at the molecular level. At the same time, microbiologists in the Philippines began to feel the need to form an organization which would promote and encourage scientific endeavors in microbiology and its related fields. Thus, on October 14, 1971, 23 professionals from different units of the University of the Philippines, Los Baños, gathered to form the Philippine Society for Microbiology. On November 23, 1971, the final draft of the Constitution was approved as certified by the following 20 signatories. To mark the creation of PSM, the first annual convention was held at the Plant Pathology Auditorium at UP Los Baños on March 4, 1972. From a charter membership of just 20 professionals, the Philippine Society for Microbiology Incorporated has grown to over 4,000 members, not only from the Philippines, but from the Asia-Pacific region as well. In the 1990s, PSM felt the need to increase and encourage participation and interaction among professionals from the academe and industry and from all over the country. Through the initiative of Professor Rasubal, PSM started holding its conventions in bigger venues. PSM also started to establish the first of its regional chapters. On February 2, 1994, the PSM Visayas chapter was established. PSM Mindanao chapter was established on May 17, 1995. Not to be left out, the PSM Northern Luzon chapter was established on March 17, 2000. More recently, PSM Bicol chapter was established on September 29, 2007. Besides the annual convention, PSM also hosts the Asia Pacific Biotechnology Congress. Held every 5 years in conjunction with the annual convention, the Congress commenced in 1986. It has attracted the participation of many international participants and has provided a venue to foster greater interaction with foreign scientists. With a vision to facilitate accreditation of qualified PSM members and to recognize individuals who have contributed exemplary work in microbiology, PSM established the Philippine Academy of Microbiology, or PAM, on May 22, 1991. To date, PAM has accredited over 165 registered microbiologists, 50 specialist microbiologists, 40 diplomates, and 20 fellows. PSM has given recognition to individuals who have excelled in microbiology and related fields. In 1985, the Most Distinguished Microbiologist Award was first awarded to Professor William Fernandez and Professor Macario Palo. In 1987, the award was given to Dr. Patrocinio Santos. In 1989, the award was given to Dr. Lydia Hoson, and in 1991, the award was given to Dr. Ruben Aspiras. Since 1987, PSM has been giving out the Outstanding Microbiologist Award. The first awardee was Dr. Ruben B. Aspiras. To date, 24 scientists have been recognized for their outstanding achievements and contributions. More recently, PSM paid tribute to Professor William L. Fernandez for launching the WLF Excellence in Teaching Microbiology Award, which honors individuals for their exemplary teaching of microbiology. The first awardee was Dr. Esperanza Cabrera of De La Salle University, Manila. PSM also recognizes institutions that have helped the society in its activities with the Institutional Service Award. Over the years, PSM Incorporated has formed important linkages with the academe and industry. In 1994, Colgate-Palmolive Philippines Incorporated launched Protax in partnership with PSM. In 1998, Procter & Gamble Philippines partnered with PSM to launch Fit Fruit and Vegetable Wash. More recently, Joy Antibac, a liquid dishwashing detergent, was also launched where several PSM board members also served as talent for its TV advertisement. In partnership with the American Society for Microbiology, PSM started granting poster awards in the different fields of microbiology. These successful partnerships have allowed PSM to carry out its various activities, as well as provide thesis reproduction and travel grants to deserving members. In 2001 and 2002, PSM conducted a nationwide seminar series on frequently asked questions about biotechnology called Biotech FAQs, which was held for teachers in Luzon, Visayas, and Mindanao. This activity was supported by DA, USDA Philippines, and ISAA in cooperation with NINBB UP Diliman. PSM recognizes the importance of increasing awareness in microbiology and biotechnology among the youth. It co-organized the first biotechnology quiz contest for high school students together with the Biotechnology Coalition of the Philippines, the National Institute of Molecular Biology and Biotechnology UP Diliman, the UP NIMBB Society, in cooperation with the PSM regional chapters, CIRCA, and various universities throughout the country. The quiz contest is the brainchild of former PSM President Dr. Cynthia Hedreda. Hundreds of high school students from all over the country participated in the 6 regional elimination rounds, which culminated in the National Biotechnology Quiz Contest held in Manila on March 8, 2008. 37 years. And 37 conventions later. PSM forges into the 21st century with the same vision of providing a forum for Filipino scientists to showcase advances in microbiology and related fields, not only locally, but through more vigorous international participation.
From the humblest of beginnings to one of the most recognized scientific organizations in the country. Let's revisit the last 5 decades of PSM to know more about its history and legacy. In 1971, 23 faculty of the University of the Philippines, Los Baños established the Philippine Society for Microbiology, That same year, its bylaws and constitution was approved by 20 signatories. In 1972, PSM held its first annual convention at the Plant Pathology Auditorium at UP Los Baños with 20 charter members. Since then, PSM has grown to over 6,000 members from the Philippines and countries in the Asia-Pacific region. To better serve its members in different parts of the country, PSM regional chapters were established in the Visayas in 1994, in Mindanao in 1995, in Northern Luzon in 2000, and in the Bicol region in 2007. To extend its reach and to foster collaboration with microbiologists in the Asia-Pacific region, PSM began hosting the Asia-Pacific Biotechnology Congress in 1986. The event is held every 5 years together with the annual convention and is attended by speakers and participants from around the world. To facilitate the accreditation of qualified PSM members and to recognize those who have contributed exemplary work in the field of microbiology, PSM established the Philippine Academy of Microbiology in 1991. As of 2020, PAM has accredited 272 registered microbiologists, 75 specialist microbiologists, 70 diplomates, and 27 fellows in its registry. Currently, PAM has been successful in endorsing the microbiology bill, which will regulate the practice of microbiology in the country. Another milestone in the history of PSM is the founding of the Microbiology Consortium, which aims to be of service by transforming microbiology education in the country by promoting new and more effective ways of teaching microbiology and excellence in ethics in the practice of microbiology as a profession by sharing resources and information among member institutions. The Microbiology Consortium was formally launched in 2018 during the 47th PSM Annual Convention through a ceremonial signing that included 31 HEI from different regions of the country. To recognize members who have excelled in microbiology and related fields, PSM has given various individual awards since 1985. The first recipients of the Most Distinguished Microbiologist Awards included Professor William Fernandez, Professor Macario Palo, Dr. Patrocinio Santos, Dr. Lydia Oson, and Dr. Ruben Aspiras.
Hello everybody, the business meeting and general assembly will start very soon. May we call the President, Dr. Howell Ho, Treasurer, Dr. Ursula Bigol, and the corresponding Secretary, Ms. Carmen Vicera, to please come on stage. Thank you.
Good afternoon. So apologies for the delay. So we will try to keep this as short as possible and as quick as possible. So can we have the... Ano ba dapat? So I would like to call to order the business meeting of the 52nd